Associate Professor from CAS in Marine Biology from the Annamalai University. So we look forward, just as we had a very resourceful day yesterday, we look forward to a very resourceful day today because our resource persons are not short of any caliber, especially in the field of aquaculture. They are people who are very experienced and robust in their fields. So we look forward to take away a lot of key points from them, especially in a department who is mainly concentrating on research in aquaculture. So I now hand over the session to Dr. Chitra, the Assistant Professor from the Department of Zoology.
Very good morning to everyone. We start today's program with God's blessings. I invite Johan Gifty from First MSc Zoology for prayer song.
You are filled with compassion and mercy and grace, with your banner of love over me. I am longing to see you one day face to face, and to be with you endlessly. Lord of lovely you are to me. Lovely Lord, you are all to me. Lovely Lord, full of purity, worthy of honor and majesty. Lord of lovely you are to me. Lord of lovely you are to me.
Thank you, Johan. I wholeheartedly invite our resource person of this first session, Dr. Charan Ravi, scientist, PMFGR Center, MFGR ICR, CMFRI campus, Kochi, Kerala. I welcome you, sir. Now I invite Abhirami Santosh from Second MSc Zoology to welcome our chief guest of the session.
Yes, ma'am. Hope is the one thing that can help us get the darkest of times. With this thoughtful quote, I wish you all a gratifying morning and genuine welcome to one and all gathered here. On the behalf of organizing team, it's my esteemed pleasure to welcome you all for today's national seminar on aquaculture production, advances and constraints. With deep pride and humility, I welcome our guest speaker for technical session 3, Dr. Charan Ravi, scientist, PMFGR Center, National Bureau of Fish Genetic Resources, CMFRA campus, Kochi, Kerala, for guiding us on the topic, augmenting conservation with aquaculture, unlocking the potential indigenous fish species for enhancing fish production in freshwater aquaculture. I'm profoundly delighted to introduce our chief guest. Sir had completed his Bachelor of Fisheries Science from College of Fisheries, Mangalore, KVAFSU, Bidar. Master of Fisheries Science degree in Aquaculture in Central Institute of Fisheries Education, Mumbai. Got Doctor of Philosophy in Aquaculture in Central Institute of Fisheries Education, Mumbai, under the title Comparative Evolution of Reproductive Performances of Asian Catfish Clarius batrachus collected from wild and captive conditions. His special skills include in the field of reproductive physiology, captive propagation of endangered species, fish milk cryopreservation, and cell culture. Sir attended various training or workshops include 11 days international training on regional capacity building program on biotechnological tools in aquatic genetic resource management and ex situ conservation by ICAR NBFGR from 7 to 18 December 2020. 4 days training on fish milk cryopreservation for genetic improvement of broodstock organized by ICAR NBFGR from 4 to 7 Feb 2020. 2 days training on demystifying interactive dashboard Xanthus Institute on June 2020. 21 days Generation Aquaculture funded by ICAR. Network Project on Ornamental Fish Breeding and Culture funded by ICAR. Establishing Germplasm Resource Center for Marine Ornamental Invertebrates, harmonizing biodiversity conservation and promoting livelihood to the islanders of Lakshadweep funded by DBT Government of India. Setting up of Ornamental Fish Village, way forward to promote to mangrove dwellers and marine biodiversity conservation at Maharashtra, funded by UNDP Mangrove Cells Government of Maharashtra. Exploration and cataloging of fish diversity from Kaveri River Basin, funded by ICAR and BFGR. Sir has following achievements on publishing 15 international and 3 national papers, 35 abstracts, 12 technical articles, and 1 book, and has attended many national and international conferences. We are so glad and deeply privileged to have such prestigious person among us, and deepest gratitude for sparing your valuable time with us, sir. Welcome you, sir. I desire to extend my grateful welcome with all reverence to our Secretary, Reverend Dr. Sister Ruby Alangaramedi, our Principal, Reverend Dr. Sister Helen, and a Vice Principal, Reverend Dr. Sister Mary Fabiola, IQAC Coordinator and Head of the Department of Zoology, Dr. Pauline Vasanthi Joseph, ma'am, and also a gracious welcome to all the eminent faculty members, renowned speakers, scholars, students of our department and from various departments for this national seminar. We are honored to have you all with us and deeply appreciate you to offer you a most grateful welcome once again. Thank you.
Thank you, Abhinami Santosh. Once again, I welcome Charan sir. Sir, over to you, sir.
Thank you, ma'am. First of all, I would like to thank Numerous College for Women for organizing this national seminar, Aquaculture Production, Advances and Constraints. And also, I would like to thank Pauline ma'am and Anil. Seminar. My voice is audible, no? I'm clear, no, ma'am? Hello?
Yes,
Hello?
sir.
Yeah. Shall I share my screen?
Yes, sir. Yes, sir. You're audible, sir.
My screen is visible, ma'am?
Yes, sir.
Yeah. Okay. The topic of my presentation is augmenting conservation with aquaculture, unlocking the potential indigenous species for enhancing fish production with freshwater aquaculture. So in this topic, are we going to talk about the fish genetic resource of our country, and I'll be briefly tell about the global production strategies as the aquaculture as well as capture fisheries and Indian context, and also I'll be going to tell the perspectives of the exotic fishes, their positive sides and the negative sides, and I'm going to talk about my core of my topic about the indigenous species and how our institute is playing role in this aspect that I'm going to elaborate also.
Sir, sorry.
Hello?
So 1 minute. Sir, please present in full screen, sir.
It's in full screen only, ma'am?
It is not in full screen, sir. Please present in full screen, sir.
So, it's in full screen only. I open in full screen. Shall I stop and again share it?
Yes, sir.
Now is it okay? Hello?
Sir, yes, sir. You can continue, sir.
Okay. The fish genetic resource consists of all fish and aquatic invertebrate genetic material which has got actual or potential value for culture and capture fisheries according to FAO. So as per the estimates and trade, There are about 62,300 vertebrate species, which includes 32,000 fish species. And out of 32,000 of fish species, 97% is bony fish and rest 3% is cartilaginous species. Cartilaginous means sharks and rays. So this figure is not the final one because the rate of discovery of new species per year is almost nearly 200 number per year. And also the global survey also indicate that there is more than 5,000 species are yet to be discovered. So one of the main mandate of our institute is exploration, characterization, and cataloging of countries with genetic resources. So in this aspect, we have a database that is Aquatic Genetic Resource Information System of India. This system, this database tells about the different aspects of native fish genetic resource reported from the Indian waters. Maybe in very soon it will be coming to a public domain, which will give all the detail about the countries with genetic resource from the different water bodies. From this database, we can see we have 3,157 kingfishers, which is belongs to 47 order, 247 family, and 1,022 genre. And out of this 3,157 species, freshwater fish is 1,039, marine is 1,982, and brackish water is 160. There is also additional exotic fishes, that is 462 number. So if you look into this figure, it is clearly tells that our country is genetically diverse and rich, and it almost represents 8% of the global finfish diversity. Now, what is the role of genetic resource for sustainable fisheries? One is that direct consumption. So, because with this diverse genetic resource, there will be more scope for species for taking in aquaculture diversification. They will provide an raw many material for the species to get adapted to a short and long-term changes in their environment. They also serve as a biological requisite for taking a genetic improvement program and that can be maintained over the long term. Now the big question is that how this abundant genetic variation can be managed, conserved, and meantime it can be exploited to reach the present and future production goals. If you look into the global stats, the global fish production is 178.5 million ton in 2018, out of which 82 million ton is coming from aquaculture, nearly 46%. And China forms the lion's share of the total production, which become nearly 35% of the total global production. That is a huge contribution from China. With the global food increasing at an annual rate of 3.1%, almost double that of annual world population growth, that is 1.6%. And compared to other animal agency, this is higher than because
Disturb you, sir.
hello.
Slides are not moving, sir. Sorry to disturb you, sir. Slides are not moving, sir.
Now is it moving?
It is in the first slide itself, sir. No, sir.
But it's going on. What I to do? Not changing.
Sir,
Hello?
once again, sir, it is your slide is not moving, sir. Once again, please present your screen, sir. Give it in the entire screen, sir.
Now?
Give it as entire screen, sir.
Okay.
When you share your screen, give it as entire screen. Not in a window, sir. Give it as entire screen.
Your enter screen, right? Present your enter screen, right?
Yes, sir. Yes, sir. Then open your slide, sir.
And now it is coming. Hello?
No, sir. No, sir. No, sir. So first open your slides, then minimize it. Then, yeah, it's coming now, sir.
Coming? Now is it
Yes, sir.
coming?
Now it's perfect, sir. Now it's perfect, sir. Give the
Okay.
side, sir.
Now slide
Side
moving.
button, sir. Yes, sir. Yes. Now it's perfect. Yes, yes. Yes, sir. You can continue.
Okay, this is about the aquatic genetic research information system of India. Uh, about the global food fish consumption is increasing at an annual rate of 3.1%, already explained. And per capita food fish consumption, which is going from 9 kg to over 20.5 kg at present, that is in 2018, nearly 1.5% per year. If you look into the graph, you can see this is a graph of global production, which includes capture and aquaculture. In that capture, you can see a red and orange bar. That is red bar is for inland capture and orange is for marine capture. And in aquaculture again, inland aquaculture and marine aquaculture. Dark blue is that inland aquaculture and light blue is that marine aquaculture. What this graph is saying is this capture sectors after 1990s, this production is not at all increasing. It is almost in the saturation level. Means this global capture fisheries is almost about 70% got exploited. We cannot expect any more further increase in the production from these sectors. Now, how do we provide food? Then, so what is the option? If we slip into the aquaculture, they keep on going trend. So, will our aquaculture will be a hope for our to fulfill the needs of a growing population? So, at present, this aquaculture is growing at a fast pace of over 8%. Nearly 600 number of cultivable species is registered in FAO state as of 2014. And in terms of global production, India stands third with a contribution of 14.16 million tons, which includes marine 3.72 and inland 10.43. The total value fishery export is 46,600 crores and quantity 12965 tons. Aquaculture India is growing at an annual growth of 6 to 7%, which is stands second in the world total aquaculture production with a contribution of 6.3 million tons in 2017 and 18. The national productivity of India stands at 3 tons per hectare per year, with per capita consumption is 8 kg. So that is the production status. If you look into the available resource, rivers and canals is 0.2 million kilometers, reservoirs 3 million hectares, tanks and ponds 2.3 million hectares, fields of gold lakes and Jalek Tota Wood is 1.3 million hectares, Brackish coated area 1.36 million hectares. So far, about 0.65 million hectares of water has been used under fish farming, which covers 1.1 million beneficiaries, which is hardly less than 10% of the available resource. So, and regarding the species, what are the species we are used in aquaculture? Is that many of the Indian major carp, that is Katla, Lady Orvita, and Chiranus Mutala, and one friend freshwater shrimp, Macrobus and Rosamur. The other species exotic fish, mainly silver carp, that is Hypothalamus molluscus, Pinopyramida nidala, common carp, that is Cyprinus scorpio, and some exotic catfish like Pangasius, Hypothalamus, Tilapia, Horeochromis, Osimutrus, and Niloticus. These are the ones which are mainly contributing of our core of our culture production. So now the question is what we have to worry about this. We have a production, we are stand second in the world. We have a production of 6.3 million tons. Then why we have to worry? Thing is that if you look into the available genetic diversity, I told in the first slide nearly we have 3,000 number of fish species. But how many number of species we are included in that culture? Hardly 3 species. That is Indian major carp. Right. So the index of biodiversity utilized for aquaculture is in the order of 0.13%. That means in that 0.213, it is 85% is coming from IMC and rest 5% from air breathing and other fishes. So it is very important that the culture of the resource which can provide a sustainable utilization is not proportionate with the level of biodiversity what we have and the agroclimatic environment what we have. The most important is that we have to go for a diversification of species in order to enhance the production and by exploring the various aquatic resources as well as agroclimatic environment. Not only this, what is the existing species we are using? All these species not at all improved strains. They are just not unimproved strains. And many of the farmers nowadays are complaining that they are not getting the production how they were earlier they are getting the production. Same way it is not. Why? Because there is no proper bush management, and that is resulting in a genetic pollution of this existing stock. And even if you talk about the ornamental species, there we have a good amount of indigenous species, but hardly we have seen of those indigenous ornamental species. Mainly, if you see any of the stock, it is mainly dominated with all exotic ornamental species. So if we think about, if we want a blue revolution or blue growth, then we have to properly manage this aquatic genetic resource. So before getting into my indigenous species, let me have a discussion about these exotic species. The exotic species or alien species which have been introduced for different reasons in our aquatic system. There can be reasons aquaculture or fisheries purpose or angling or sport purpose, sometimes accidentally with a ballast water or some other water source it might have come, some ornamental purpose and unknown and other reasons. So during 1950s, 2000, there are 14 exotic species that have been introduced in India, namely common carp, silver carp, grass carp, salmon is like three, Salvelinus continentalis, Oncorhynchus negra, salmonella, brigade carp, tilapia, Orocomis landoticus and Mosambicus, African catfish, Laris garatinus, red-bellied papu, Erectus brachypomus, Malaysian catfish, Pangasiran hypothalamus and white bone, Leptophilus fundamental. This why I highlighted in these two species is because this has been introduced illegally into the system. Then later this papu has been permitted by government. So ornamental industry thrive well in India. They are mainly dominated with exotic species itself. Since and that has been introduced by the fish office from different parts of the world, and there is no clear information is available about from where has been introduced and when and all. So as of now, nearly 200 exotic or aquarium fishes are there, which have been cultured and breeding is going on throughout the country. So the photos, what you can see is all these exotic fish. If you go to any shop, you can see any of the indigenous ornamental fish. Whatever you see, maximum will be 90% will be exotic one itself. You can see angel, guppy, molly, discus, gorami, arowana, sucker catfish, alligator catfish. Now, the question is that whether this exotic fish is good or bad. So, these are some. I have made some arguments about this aspect. If you can, the good part you can see it's a global view about 1,155 species introduced for aquaculture. Only 6.8% and 0.7% are considered ecological and socio-economically adverse, and but 4.5% and 24.5% were considered beneficial. So the positive side is very more here. And most aquaculturally important alien species like Mosambicus and Niloticus, which is contribute to the inland fish production in reservoirs and destination countries. If you see any of the reservoirs, if you go and see, there is no reservoirs or water body without this tilapia. In India also. And this introduced fish is recognized as one of the most effective fisheries management tools for increasing the production in the inland water. The production of wanami in China comprises almost 70% of the country's total marine shrimp production. If you talk about the Indian context, about the exotic fish, they are contributing more than 37% of aquaculture production in India. It is not only now, it's since 1990s. The major species which is contributing in aquaculture production of India is that silver carp, Pangasiran hypothalamus, Leptophilus wanami, and common carp. And there are enormous collections of tilapia coming from the different reservoirs which are not being recorded officially. And not only this, there are many ornamental fishes out there. Since they are very easy to breed and raise, it has been very well adapted in our system also. If you look at here is the pie diagram, you can see that silver carp, how these exotic fishes are contributing what I call 37%. And in the shrimp sector, you can see the more than 50% of the production is now coming from the exotic shrimp. That is wanami. After the introduction of wanami, there is a huge boom in the production sectors. So why we are easily getting attracted to the exotic fish? One is that availability of the seed in any number, and the practice of culture is a very design practice for these species are available, and you can see the development or availability of a SPF, so that is specific pathogen free. If you look at the fish, the major problem is the disease. I think tiger prawn, which is the WSSP. Now, Vanami we are getting that is a WSSP stock. So that is one reason. Then they have a capability to survive in a harsh adverse conditions. They can tolerate that good any water quality parameter also. Today we are able to culture Vanami about 2,500 hectare area and using approximately 1,500 tons per hectare per year. So in the negative aspect, the major problem with introduced with exotic species is the disease. In fish, mainly the UVS, which is causing a major problem in India. And other than this is norovirus and koi herpes virus, which is recently identified and that has become spreading in many of the cyprinids also. And in few of the viruses like WSSC, IHSNA, IMNE, Taurus virus, yellow red virus, EMS. And about the disease part, I think the next lecture will be detailedly discussed about that. The other problem of exotic species is that, as per the FIS stage, there is 27 species of exotic species that have been reported by one or more countries having adverse ecological impact. So many of these impacts has been not reported in most of the literatures. We don't have exact data and how these exotic species are affecting in the native fish. And most of the changes what is happening with this introduced species, the habitat destruction and environmental changes for this native species because of the alteration in their food web and transmission of the pathogen also. Though some of the permission is given for culture, these exotic species in captive condition and control condition, but many of the times these fish may get escaped into the natural ocean. For example, before 2 years before, there is a flood was happened in Kerala that has led to many of the farms got flooded and most of the this papu which is an exotic which are all got introduced into the different river system of Kerala. You can see this Chalapudi river they are getting catch and this African catfish which have got well established in most of the river system of India. So apart from that, there's a negative side on genetical aspect. They have the capability of getting hybridized with our native species. Sometimes they produce infertile hybrids, sometimes they produce fertile offspring, but will ultimately result in the pollution in the gene pool of the native species. Environmental aspect involves reducing the biodiversity or decreasing the number of endangered species by destabilizing the ecosystem. Some economic impact like there will be reduction in the fishery productivity. Though these exotic fishes may lead to a good production, but the amount of benefit or profit is coming from the exotic fish is less compared to the native fish. There was a study in Jainspur Lake of Rajasthan where tilapia is established accidentally and so tilapia totally reaches IMC in the fish production. This has led to the reduced income to the farmers. These are some of the exotic pathogens got introduced in India, like UES, Trichodina reticulata, Triparatella, Foxy, and White. This is how these pathogens are called exotic species, like virus 42%, 4% is the mixed infection, and bacterial as well as fungal infections, all coming from foreign pathogens. Now the question is what can be done with this exotic? It is not to ban these alien species or to abandon the regulation to their movement, but rather, as stated in ICES and the CBD, to assess the risk and benefit associated with their use and then, if appropriate, develop and implement a plan for the responsible use. And there is a need of ecosystem-based approach. So where it has to be introduced and how it has to be introduced. And after introducing a strong measure is to be required in order to monitor this introduced species. And also we have to have a balance. We have to weigh benefits. How is it benefit? It is really more benefit in the development and in terms of economy of the country, but it's not the cost of at the cost of our diversity. So I was telling about this the exotic fish bringing diseases and all. So there are in order to safeguard our indigenous species, steps have been initiated for upgradation of quarantine and disease diagnostic facilities and expertise. And Endanger initiated a program, national surveillance program for aquatic animal disease in 16 states and 2 union territories, with 27 collaborating center for monitoring and control of spread of national and international concerned diseases. So this, because of this program, we can able to our diagnostic capability of state is also got strengthened, and we could able to do the surveillance for oil-listed diseases. This this is the two document has been ended released. That is a guideline for import of exotic fishes in India. Now let me come to the core of our topic. That is indigenous fish. Now, what is the importance of our indigenous fish? Why we have to why we have to go towards more of indigenous fish? Because we have seen our country is blessed with a good amount of native fishes, which supports capture fisheries in such high price also. But many of these populations got declined in the wild because of continuous effort towards catching of fish, habitat modification, and some climate change and so on. And many of them are fast growing, but that species has been neglected in order to introduce into the culture system. But the growth of the fish may not be as same as IMC or Chinese crab. If you think about the price, what IMC fetches, for example, it fetches 150 rupees per kilo, but these fish can fetches 200, 250 rupees, far better price than the IMC also. So this conserving indigenous species are more important for to maintain the ecological equilibrium as well as the economic gain from the capture resources. So productivity enhancement through harnessing domestication and cultural patterns such as the use of equal priority, and developing a strategy that harmonizes both the priorities could serve a good purpose, particularly for the region and in general for the nations that are restored with biological wealth, but are also staying with a large population quickly. So, developing the captive breeding technology for this indigenous local species and bringing them into aquaculture, it will help not only in conservation but also contribute to the food and economic security of farmers. India became a signatory in the region-specific aquaculture in the Summit 1992, and one of the recommendations in this is to use native species wherever possible for aquaculture purpose. The concept is mainly to widen the food basket with a species of regional preference and not to replace or substitute IMC. So, tackling the issue of increasing disease threat and conservation measures of several threatened endemic species, meeting the emerging demand for species with a short production cycle and bringing high-value species under diversified farming systems. So, with this aspect, many of the ICR Research Institute developed the breeding and seed production technology for more than 60 species of indigenous species. So here I'm going to tell about the important species. I will mainly restrict my talk to the species which are endemic to the Western Ghats and southern India. I am not going to tell all indigenous species of the country. The first species is Echinobarbus carnaticus. This species have ability to reach a large size as 12 kg, and they have feeding nature with herbivorous. They can also macropod feeder also. They are found distributed in Karnataka, Kerala, and Tamil Nadu. So what actually gap about this species is that the culture aspect is detailed. So the breeding technology has been developed by SIPA. Now still in the farm evolution is going on about this species. Echinobarbus microcogon, which are able to reach up to 5 kg, and the animals of this species also have an ornamental demand also. They are well distributed in Kerala and Karnataka. But about this biology of this species, there is no information is available, and there also exhibit a taxonomic ambiguity. and this species has been considered as critically endangered. So there is a more important to work on this species to bring on conservation as well as to evaluate for culture purposes. Echinobarbus culturals, it's a microphyte feeder. It can reach up to 3 to 5 kg. They are found distributed in Karnataka and Andhra Pradesh. The breeding technology has been initiated by SIPA. Still, there is a taxonomic ambiguity existing in this species, and the culture aspect of the species that need to be studied. Ipsilabaga duckling, they can grow up to 10 kg, which are found distributed in Kerala, Tamil Nadu, and Karnataka. There is no information about their reproductive biology, and there is just a taxonomic ambiguity and culture aspect is to be studied in detail. Istamus sarana. So these species are, though you can see most of the riverine system with these species are throughout India it's available, and there have been this breeding technology of this species has been worked out, and main thing is that still the culture aspect is not being studied in order to bring it to farmers. Leberusmari, this one species which are endemic to Western Ghats, mainly which has got a very good demand in Kerala. Though the captive breeding and captive protocol has been developed by NBFCR, the present work on monoculture and polyculture aspect is going on about this species. Uranus cirrhosis, which is distributed in Tamil Nadu and Kerala. There is no information available about this species in terms of culture as well as reproductive biology, and totally many more work has been done on this species, which has got a potential to grow to a bigger size. Labia pontius, same. Monoproctopus carmuca, which is found distributed in Kerala and Karnataka. Osteocalypsis longidorsalis. Neolithicalis winadensis. And so one Mahashi, where it can grow up to 4 kg, and that has got an ornamental value also. The biology of this fish has not been studied, and no breeding protocol also not been studied. So still Clarius decimary. This is one of the catfish which is endemic to Western Ghats. Now the present breeding work has been standardized and engaged here. The culture aspect is going on. Horobacus brachytoma. This fish is yellow catfish, endemic to the Western Ghats. Very good demand is in Kerala. Commonly called as maniapuri. Ectoplus suratensis, which having a very good market value, nearly plus 300 to 400 rupees, and mainly have very high demand, which is there in Kerala. Though most of the breeding and culture aspect has been studied, culture, RS, and now at present you can see the species in culture also, in cage culture, and as well as biofloc system also is coming. Apart from that, there are many other species like Labiopimulators, Labioculbers, Labioportalis, Ipsilocorpus thomas, Ipsilocorpus polus, Ipsilocorpus dubius, which are capable to grow a larger size. But there is a more study required on this species to bring into the culture aspect. The catfishes like Sparata aerodes, Collabas, Opa Vernaculators, Fungaceous, and some Sanna species like Channa Murali, Channa Pancata, Channa Saeta, Channa Diplogramma. Now, apart from the culture aspect, NGFL is working on conservation of this species also, native indigenous species. The concept, innovative concept has been developed by declaring India state fish, where 60 state of the country become the partner with NGFL, mainly to develop a strategies for conservation and enhancement of this selected state fish. And also, this has been observed a very fruitful result through this concept also. Live gene bank, where endangered endemic species can be maintained in near natural conditions, which can help to recover and utilization of genetic diversity and can be used in conservation program and genetic enhancement. More regionalizing in different agroclimatic zones are proposed in collaborative mode to accommodate more species and develop some of the repository into this parks or ecotourism zones to create a mass awareness. These are some of the lightning germplasm research center established by NBFCR for on-farm evaluation of genetic resource. You can see in NBFC Lucknow, there is a three species IMC and the Claris Magur Ompa Vermaculatus, Airoli Mumbai, Marine Ornamentals, and female jet Center Kerala. There are four species, Claris Discipline, Endocarpus Punctatus, Saurobratus Brachysoma, Agati Laxity, mainly ornamental invertebrates. Nagarjuna Sagar, two species, Ompa Vermaculatus, Fungaceous Silesi, and in Manipur and Gavati. So these has been taken as a working model mainly to establish the breeding population of these indigenous species and for captive propagation, mainly to aid in conservation and childhood promotion. Now, what we are actually doing in Kochi, we have in the Jamkasam Research Center, it is uh, mainly to hold a multi-species of groups of theotypes for conserved and captive propagation. This will serve the facility for on-farm evaluation and characterization of indigenous species and their genetic stock and center for capacity building for farmers and Department of Fisheries. Here, under the consortium project of agrobiodiversity, they are taking up the conservation and captive propagation of four endangered species, mainly Claris disumeri, yellow catfish, label disumeri, and Hemibarbus truncata. The Claris disumeri is commonly known as Nada Mutti. The main problem confusion comes with the African catfish. The picture you can see the occipital process, how the V shape in the case of head and which is not which is like a U shape in the case of disumeri. This is how we are doing the breeding and seed production. The interesting thing about this species is the possibility of an induced natural cloning. Other catfishes like Claris magura or African catfish, where need sacrificing the male by cutting opening the belly and taking the male to make the sperm suspension. But in this species, it is possible to they will respond after injecting. They will naturally they release the milk and eggs to fertilize the eggs. And one of the most advantage of this is the growth potential. You can see in the 8 months they are able to attain the average growth of 250 grams with the maximum growth of 400 grams. This is one of the it is a very good result what we observed. So still we are evaluating this fish in different farm system to make sure that the growth is we have got a good growth potential so that we can distribute the seeds to the farmers. And the next species is Brachysoma. So, commonly known as Manipuri, ICR and the joint with RS Kumarkam has developed the breeding and diversification protocol, and also the stock-specific breeding assisted river ranching was also done in the Vembanas Lake and adjacent rivers for this species also. At present, we are upscaling the seed production of this species. You can see the pictures where the fruiters fertilize eggs and stocking in protein up and even the distribution of fingerlings to the farmers as well as the ornamental entrepreneurs. The best part is that this fish have got both the potential as a food fish as well as ornamental value also. The label is similar. At present, we are producing the seed and we are distributing the seed of this label similar to the farmers as well as state department officials, mainly to build the food stock of this fish because these fishes are hardly in wild. It's hardly difficult to find these animals because of their numbers getting reduced. With our program, we are able to successfully breed and multiply these species and distribute the seed to the farmers, and also we are doing the ranching in different water body also. This is one of the catfish which are critically endangered. They are known as it is the Hemibarbus punctatus. It is endemic to the river Kaveri. One of the objectives of our work in agriculture is exploration. We have to do in order to document the opportunity because we have to explore the different water body of the country. So we are mainly doing the southern India. So when we doing the exploration, we are coming across this species. And when we got some few individuals, we brought it to our captivity, we studied biology, and we are able to successfully feed this fish. Now the seed production is going on. So another species is a new species, which is described by NBS here. The name has been given as Fungaceous silesi. And this fish has been a neutral profiling has been studied along with the Fungacidon hypothalamus, one of the exotic catfish. And we have been observed that It is nutritionally superior than the hypothalamus. So by looking into this, we have started developing the brood stock, and also we observed the growth of this fish, and we have which has noted a remarkable result in the growth. So very soon we'll be bringing this fish to the culture also. This is how we are monitoring the growth of the fish. Individual fish, we will be tagging them, and by using a stick tag, that is fashion integrated chip. With this, each fish how much is going, we are able to record it also. We are also documenting the embryological stages of these different indigenous fishes. Uh, how this, apart from this stock enhancement to ranching, the captive breeding and stocking in wild to compensate the declining fish population. As we know, there is a good technologies available for IMC, that is Indian Major Cup, but many of the indigenous fishes, which is having the commercial value, are yet to develop the technology. So there is a captive breeding and naval rearing of non-conventional fishes like Chitala, Wompak, labeled Rusumari, and 15 indigenous ornamental fishes have been developed under NATP project, and that has been a remarkable success also. And there is also mass-scale seed production and ranching of these species need to be done for in situ conservation of these species in natural water. This is one of the successful story what we have done with the hello catfish in neighborhood summary. After the seed production, we are stopped in their native river, and that has resulted in the increase in the landings from 1.8 to 11% increase of horobragus, and 0.68% to 39% in the case of neighborhood summary. Now, community-based aquaculture. Aquaculture may be more effective in conserving resources than regulation that restrict exploitation. So, for example, in marine ornamental sectors, the absence of alternative livelihood, restriction on the collection or trade target species often lead to targeting a new species that are not able to withstand the extension. In such context, developing a concept of community-based aquaculture that will be very helpful for shifting the resource to resource. On this idea, we have a project that is setting up now marine ornamental fish village partnership. The way forward to promote the allowance to mangrove wellness and biodiversity conservation. I think yesterday this we have been understanding about this concept of this project to establish a model ornamental fish village. Then I already in the first slide I told whatever the species we have, it's not an improved size. There, what will happen? For example, INC in the major cup, in the farmers are culturing, what he will do, whatever the fishes which are grown, very good size of fishes, he will be selling for good rate. But whatever the small fish, he will keep it, and that will develop as a food stock. So that will be when you using continuously that fish, the end one which is coming from the smaller fishes is the quality of the lesser growth also. That will replace coming generation that will have a more negative impact. So that is resulting in a negative selection. So here how we can avoid the negative selection. These are some table I have given for each fish. What should be the minimum age and what should be the minimum weight for taking up the breeding activity. Now there is also an improved strain which is developed by SIFA, that is giant rohu, which has shown almost 70% higher genetic gain compared to the normal rohu. And also this they also shorten the culture period from 8 months to 2 months. They also now to again, they can tolerate the infection of Aromonas hydrophila also. And the production of almost from 10 ton per hectare was increased to 20 ton per hectare per year using the giant row also. Same line of work is also going on with Katla and Macrobacter mosambasi. So there are called inbreeding depression. What farmers are doing means when they're not maintaining the records of pedigree for breeding eggs. For example, if they breed one lady, and the next after the selling the left out seed, they'll be developing the brood stock, and next year they'll be using the say young ones from that brood stock for a brooders. So that will result in breeding among the brothers and sisters, which will result in the quality of the which will get degraded. So it is very difficult for the farmers to maintain the detail of their brooders or pedigree. Or other thing option is that every time they have to go to the river to exchange to get the wild brooders, or they have to get it exchange from the different hatcheries. We have developed one sound banking where females will be from the hatchery and milk will prior to the milk will be taking in fertilizer which are prior to the milk were collected from the wild brooders that will be exchanged that will be mixed with the farm females of this hatchery so that the genetic variation will be maintained. Almost some 18 farms of throughout India have been carried out of this work. Still the work is going on, mainly the genetic upgradation of the IMC. Now, looking into the indigenous ornamental fish, you can see they are diversity of fishes which are having a good ornamental value. So these are the fishes what we have collected from during our exploration. The few pictures have been shared here, and which have got a very good ornamental demand. Nearly $10 to $15 also fishes. So the major western Ghats species that are exported from India is that like Sayadya, Danisoni, Chalapudi, Dokinsia, Tamrapalli, Dokinsia exlomata, Dokinsia rubrotinctus. So many of the for many of the ornamental indigenous species, the breeding technology was developed under the NATP project. Mainly, you can see it is Labuca fasciata, Sayadya, Danisoni, Katia popodensis, and Dystophis marginensis. And at present, under the project of network project on ornamental fish breeding and culture of indigenous fishes, we have developed a breeding protocol for 3 indigenous ornamental fish, that is Dokinsia rubrotinctus, Katia septinae, and Katia negripinna. And we have also started supplying the seeds to the ornamental entrepreneurs in order to promote these indigenous ornamental fishes. So with this, I would like to conclude my topic with so before introducing any new species, whether indigenous or exotic, people local regional preference may have to be considered so that there won't be any problem of marketing. For example, in Kerala, there's a there's a more preference for Atroplus, Suratensis, and Maras. So when we take up this species here, we cannot we do not worry about any marketing of this species, and also that's a very good value also. Among all the native fish species produced locally, they are consumed locally itself. So there's a short supply chain with the fewer middlemen, which is resulting in higher returns for the farmers. Native species and varieties are known for better adapted to the local regulation than the exotic ones. And also the native species are often high local demand as they are considered locally delicacy and fetching a higher price for the farmers. Some native species surprisingly play a large role in securing access to animal protein and micronutrients in rural communities. You can see some of these local species are vital to the livelihood and food security of the lower class people because access to such species is costly and open to everyone. The catching, culture, processing, and selling of these local indigenous species are exclusively done by women, giving them the opportunity to independently sustain themselves. This potentially empowers the women should also be promoted for sports and culture of native species. Prioritizing the culture of native species by protecting and preserving the local varieties can definitely be a key solution to continue safeguarding the food security for our growing population. Thank you.
Sir. Thank you, sir.
Yeah.
Sir, I have one question, sir.
Yes,
Sir, in your CMA campus, are you doing on cultivar fishes? The research is going on in cultivar fishes or any work related on value-added products, sir?
In our center, we are working on cultivable fishes, not value-added products. There is a value-added product. There is a separate institute, CEFT, Central Institute of Fish. processing technology.
Oh.
There, it is also in Kochi only. It's located in Wellington Island.
Yes. Sir, one more simple question, sir.
Yes, ma'am.
When we are doing research work in our lab, sir, for UG or PG students, we are procuring ornamental fishes from the ordinary ornamental fish shop only, sir.
Yes, ma'am.
But it's very tough task to acclimatize these ornamental fishes in our lab, and even in the home also.
So, what actually species generally you are getting, ma'am?
Sir,
No, what species generally you are getting, ma'am?
we are getting under the ornamental fish shop. Commonly, we are getting molly, goldfishes. Yes,
These
and
fishes should not be a problem.
single fishes.
They should be managed. I don't know why it is happening there. Maybe some water quality you have been checked in your lab, ma'am. Especially if you are yes, ma'am. If you are using this tap water and all.
Yes, sir, ordinary tap water.
Yeah, then there will be issue of chlorine and all. This
Yes.
kind of checking to that thing.
But we are monitoring that, sir. Even we are very trouble to sustain these fishes.
Should not be, ma'am, because these fishes libraries are very easy to maintain compared to other indigenous fishes. It should be very something. Is it coming any disease and all, or any symptoms it is showing?
Sir, I couldn't.
Hello.
Yes, sir. Yes, sir.
Yeah. Any symptoms of disease and all showing, or anything you can observe in their dying?
No, sir. When we are procuring, they are healthy only. We are not observing any disease externally. So many times we tried, but we couldn't.
Let's check the water pollution. Mainly this water pollution is this alternative
Water quality is-
pH you can check. These two factors just check it. Otherwise, there won't be any problem with library. And when I keep some plants like that will be helpful for that.
Thank you, sir. Now I request Steffi Mary Thomas from Second Dimension Zoology to propose water tanks.
Am I audible, ma'am?
Yes, yes.
A very good morning to all of you present here. Gratitude shapes our attitude, which turns us out to be better humans. First of all, I thank God Almighty for making this session a resounding success. I deem it a great honor to propose the vote of thanks for this educative session. It's an immense pleasure for me to extend my sincere thanks to our resource person for this technical session, Dr. R. Charan, PMFGR Center, National Bureau of Fish Genetic Resources, CMFRI Campus Kochi, for enlightening us on the topic, augmenting conservation with aquaculture, unlocking the potential indigenous fish species for enhancing fish production in freshwater aquaculture. Sir, your talk gave us a lot of information on the wide variety of the exotic species as well as their useful aspects and effects, also on the importance of indigenous varieties and their contribution for a balanced ecosystem, and overall on the criteria for aquatic genetic resource management. Thank you, sir. It really was a very beneficial session. I would like to extend my heartfelt thanks to our Secretary, Reverend Dr. Sister Ruby Alangara Mary, Principal, Reverend Dr. Sister Helen, for their ardent support, and the organizing committee, Vice Principal, Reverend Dr. Sister Mary Fabiola, IQAC Coordinator and Head of the Department of Zoology, Dr. Pauline Vasanti Joseph, and our faculty members for their sincere efforts in organizing this program. Also, my deepest appreciation to each and every participant for their coordination throughout the session. Once again, I thank you all.
Thank you, ma'am. Thank you very much.
Yes, thank you very much, Steffi. Uh, Dr. Charan Ravi, I would like to thank you from the bottom of my heart. Your slides were as exotic as the exotic visuals you had explained. So thank you so much. It would have kept the interest and focus of our students. Otherwise, it's very hard to keep them for more than 30 minutes in one platform. Thank you so much, Dr. Charan Ravi. Now we move on to the second technical session, and we have Dr. Arun Sudhakar. And sir, we are so happy to have you. It's a privilege to have scientists, young scientists like you, and you are actually a source of motivation to our students. So I'm so happy that you have given your consent and that you will be talking and staying with us for an hour. All right, sir. I'll just hand on the podium to Dr. Rosalind Mary, the Associate Professor from the Department.
Thank you.
Good morning, sir.
Good morning.
Welcome to our seminar, sir.
Thank you.
Dr. Arun Sudhakar, scientist, ICAR, National Bureau of Fish Genetic Resources, Kochi, Kerala. Welcome you, sir. And he will speak on the topic present status of emerging diseases in India, in Indian aquaculture. I welcome Ms. Jagan Gipti of First MSc Zoology to give the welcome address.
Give thanks to the Lord, for his steadfast love endures forever. 2 Chronicles 2021. A very warm welcome to one and all gathered here for the technical session 4 on the topic Present Status of Emerging Diseases in Indian Aquaculture. I'm profoundly delighted to take this opportunity to introduce the guest speaker of this session, Dr. Arun Sudhakar, scientist, ICAR, National Bureau of Fish Genetic Resources, CMFRI Campus, Kochi, Kerala. Sir has completed his Masters in Fishery Science in the year 2011. He had completed his PhD in the year 2020 on the topic Visualization of Immune Response of Trout using RNA sequence technology during proliferative kidney diseases from the University of Veterinary Medicine, Vienna, Austria. He has an impeccable work profile as a scientist in highly esteemed institution since Jan 2014 till date. Sir has received Dr. H.P.C. Shetty Gold Medal as the Best Fishery Graduate of India in the year 2009 and a letter of appreciation from the Honorable Chief Minister of Punjab, Sri Prakash Singh Badla, for the research demonstration and the extensive activities carried out during the year 2016. He has also received many fellowships and grants from different agencies. Sir has published 11 papers in various journals. To name a few: Kinetics of Local Systemic Immune Cell Response in Whirling Disease Infection and Resistance in Rainbow Trout in the year 2019, Identification and Expression Profiling of Toll-like Receptors of Brown Trout during Proliferative Kidney Disease in the year 2020, Co-infection of infectious hypodermal and hemopoietic necrosis virus and white spot syndrome virus in the wild crustaceans of Andaman and Nicobar Archipelago, India in the year 2021. And he has also contributed his knowledge into various articles, books, magazines, etc., for the welfare of the students' community. Thank you, sir. You're such an inspiration to us. His current area of research is fish immune response and host pathogen interaction. It's indeed a great honor and privilege to have such a renowned personality among us. I welcome you, sir. I also extend my welcome to our secretary, Dr. Sister Ruby Alvarameri, our principal, Reverend Dr. Sister Helen, our vice principal, Reverend Dr. Sister Mary Fabiola, our IQAC coordinator and head of the Department of Zoology, Dr. Pauline Vasanti Joseph, and our dear faculty members. Last but not the least, I would like to welcome our dear participants for this session. Once again, I welcome you all. Thank you.
Thanks for
Johan. Sir, the session is open to you, sir.
Okay.
You can take the podium. Thank
Yeah.
you, sir.
And thank you so much for the warm welcome.
Yes.
Am I audible?
Yes, sir.
Good morning, everyone. First of all, I'm thankful to the organizers of this national seminar for inviting me. And today I will be presenting an important topic in aquaculture, that is present status of emerging diseases in Indian aquaculture. So my talk will be on diseases, like the new emerging diseases in Indian aquaculture, and we'll be discussing about what are the problems we are facing and what is the reason for these emerging diseases and how can we control them and what are the research efforts we have to make to tackle these emerging diseases. So I have divided my presentation into 9 parts. Like the first part will be covering the relationship between aquaculture and disease, and the second will be the fish diseases, the emerging fish diseases, and special focus will be given upon the recent emerging diseases of fish. And third will be shrimp diseases. So shrimp aquaculture is also plays a major part in the Indian system because it brings us a lot of foreign revenue, particularly by means of export. So we will be discussing about the emerging shrimp diseases. And the fourth, we will be discussing about a project, which is a national project, the National Surveillance Program for Aquatic Animal Diseases, also called as NASPAD, which was started in 2013 to screen emerging diseases in India. It's a very big project. It's a pan-India project, which is still running. And fifth, we will be discussing about the tools used in the disease diagnosis. And then we'll be discussing about the factors influencing the emergence of aquatic diseases. Like what are the factors, like what kind of environmental parameters or what kind of anthropogenic activities influence these emerging diseases. And seventh, we'll be discussing about the research gaps in the research gaps in this area. And eighth, we'll be discussing about the research collaboration needed to tackle this problem. And at the last, we'll be discussing about the prevention and control strategies. So first, we'll be discussing about aquaculture and diseases. So for the past two days, you might have seen this graph from the other fellow speakers, that is the world fisheries production, which combines both aquaculture as well as fisheries. If you see the graph, you can see a steady trend in the capture fisheries. The yellow or the orange label is the one which denotes the capture fisheries. You could see from 1990 onwards, you could see a steady production rate for capture fisheries. However, in case of aquaculture, you can see a steady growth. It's in a growing trend. So the world fishery production is about 178.5 million tons. And if you see the proportion of the production contributed by capture and culture fisheries is almost 50-50. It's not exactly 50-50, however, it's almost 50-50. So from this, we can understand that year after year, aquaculture is increasing. It has a growing trend. Similarly, if you see the aquaculture production in India, from 2010, you can see a steady increase in the aquaculture production. And by 2017, it was 6.18 million tons, and I think presently it should be more than 7 million tons. So, and if you see the Indian aquaculture, mostly it is contributed by the freshwater fishes. And this is a.
Please, sir,
hello.
Please press the hide button, sir.
Okay. Okay.
Thank you, sir.
Yeah. This graph denotes the trade in rupees, crore rupees, for the export of fish and fishery products from India. Even if you see the export by value, it is increasing year after year. There is a steady growth in the export of our fish and fishery products to foreign countries. If you see in 2019-20 financial year, we have exported worth of 46.6 thousand crore rupees worth of fishery products. So now the question is, what could be the reason for this increase in production and export? So the main reason is after globalization, like it started in early 90s, we have actually increased our, we have improved our production technologies. We have intensified our aquaculture activities. Along with that, we have also diversified our aquaculture, particularly diversification in terms of species diversification. We have adopted new species from abroad to like pangasius, and in case of shrimp, we have taken up Lithopenes vanami, which was originally from Pacific region. So this adoption of new technologies as well as diversification of species have increased the production of aquaculture. At the same time, recently a lot of new technologies are coming, like biofloc technology or recirculatory aquaculture system. So we can expect more aquaculture production in the coming years. So at the same time, our demand for protein has increased. It is expected that by 2015, we need 70% more protein that we consume today. Similarly, at the same time, right now we are losing 20% of the animal protein because of animal diseases. It's not like this data is not just for fishery products. It includes all animal products. So there is a relationship between intensification of aquaculture versus disease outbreaks. So that if you see the relationship, it is a directly proportional relationship. So when we intensify our aquaculture, when the production, our production increases. So when the production increases, we can expect more disease outbreaks. So it is directly proportional. So in the coming days, when it is expected that aquaculture will intensify further with the new technology, so we can expect more emerging diseases, or we have to invest a lot of research on these emerging diseases. So in terms of economy, like it is estimated that due to aquaculture diseases, the world is losing about $6 billion per year. So that is around 4.5 lakh crore rupees per year. And there was a study done in 2006-8, and it is estimated that in India alone, we are losing 1,000 crore rupees per year due to diseases in shrimp aquaculture. So this was 13 years back. It should be much higher now. So in aquaculture, we have the disease can be caused by various causative agents. It can be a bacterial disease or a viral disease, or it can be a parasitic disease, a fungal disease, or sometimes it could be a nutritional deficiency or a metabolic disorder. So controlling nutritional disease or a metabolic disorder is it is like comparatively easy than pathogenic diseases. So so the disease triangle. So you can say call this as a disease triangle. So for a host to get infected, three parameters are mainly needed. First, the susceptible host. The second is the pathogen, and third is the conducive environment. So if all these three conditions comes together, it will cause a disease. So how can we explain or define an emerging disease? Like how do you define it? So it's like emerging diseases can be defined as a new disease. It can be a new pathogen, or it can be a new presentation of a known disease, maybe a mutant variety of a already existing pathogen, or a new strain of an already existing pathogen, or it can be an old pathogen which is appearing in a new geographical area. So we can classify emerging diseases under any three of under these three categories. So for the past 20 years, emerging diseases have become an increasingly important area of research, not only in aquaculture or in fisheries. It has been given more focus in humans as well as veterinary medicine. So this this represents the timeline of outbreak of exotic animal diseases in India, particularly with in case of aquaculture. So the top ones are the brachyshorter shrimps or the brachyshorter shrimp and fish pathogens, and the bottom ones are freshwater shrimp and fish pathogens. We got like you can see, like most of these diseases are viral diseases, like MB, WSSV, IHHNV. They are mostly viral diseases, and we have few parasitic diseases like EHP. And if you see, most of these new viruses have been discovered in the late part of after 2010, because this happened because of the NASPAD project. So after the introduction of this NASPAD, National Surveillance Project, labs across India started screening for exotic pathogens, and we could find a lot of new pathogens which were not existing before in India. So we will be seeing deep into these pathogens in the coming slides. So, and most of these pathogens come under OAE listed pathogens. Like, what is OAE? OAE is World Organization for Animal Health. So its headquarters is located in Paris. It is an intergovernmental organization which supports and promotes animal disease control. Particularly, they focus on epizootic diseases and transboundary diseases in animals. It has 182 member countries. It publishes list of animal diseases which are quite important and notable, and research focus should be given upon. And if you see, most of these pathogens present in this slide belongs to OA listed pathogens. So next, the second part of this presentation will be on fish diseases. So the first disease is epizootic ulcerative syndrome. So what is the story, how it came to India? So in May 1988, there was an outbreak of some unknown disease in Tripura, in northeastern part of India. So people were wondering what's happening. Like, a lot of fishes were dying in wild as well as in aquaculture ponds. So soon after that, like within a year, it got spread to other parts of India, almost every state of India. It reached even up to Kerala and Tamil Nadu. So people, like, it was long back, and during that time, proper diagnostic facilities were not available. And initially, farmers started shutting down their aquaculture farms, and people were not preferring to eat fish. And even the government was advertising not to eat fish during that time. So so there was a huge economic loss and a livelihood loss for a lot of marginal people. And later, this disease was identified as Aphanomyces invadens. It is a oomycete, so it's a fungal infection. And you could see a lot of ulcers on the affected fish. And people were not preferring to eat fish during this outbreak. So this was caused by, as I said before, it was caused by a oomycete, Aphanomyces invadens. It was infecting both freshwater fish as well as estuarine fish. And OAE listed this, notified this pathogen as an important pathogen. And it mainly occurs during cold seasons, particularly when the water temperature drops below 18 to 22 degrees Celsius. And it is quite common during the winter seasons in North India. And the most important thing is it has a wide host range. It can affect a large group of fish, like around 76 species. And the transmission is mainly horizontal transmission. Horizontal transmission means fish to fish. It won't transmit from parent to offspring. It can transmit from fish to fish. And right now, many people in northern part of India use Cifax as a treatment for this aphanomyces infection. Then the second thing, second pathogen I would like to introduce is carp edema virus. So it is also called as koi sleeping disease, and it's quite common in common carp and koi carp. So it can affect both food fish as well as ornamental fish, and it can cause mortality up to 100%. It's a major threat for common carp and koi carp industry. This virus, it is a quite old virus. It was first detected in 1970s in Japan. However, after the NASPAT project, it was recently found in India in 2016. So in India, it is considered as an emerging disease. So it is a double-stranded DNA virus. It resembles to that of poxviridae, and it's 416 to 450 nanometer, and it's and the mature viron ranges between 300 to 400 nanometer in there. And if you see this virus or this disease, it's quite temperature dependent. So normally when at lower temperatures, the infection outbreak happens, and when the temperature is increased to 28 degrees Celsius, normally the mortality and morbidity of the fish used to get reduced. So we used to get a lot of these problems in the higher altitudes, particularly the fish farms from Ooty or Kodaikanal. They used to get these problems, but when they bring the fish down to the lower altitudes, some of the fishes used to get recovered. And this infection causes immunosuppression in the host that leads to other secondary infections like co-infections of bacteria or ectoparasitic infection. And in this picture, you can see like a necrotic gill in the koi carp gill, and as well as morbid fishes lying down at the bottom of the tank. And the next virus I would like to introduce is viral nervous necrosis virus. So it is also called as viral encephalopathy and retinopathy. It is an OAE list. It is listed as emerging disease by OAE. It can infect marine brackish water and freshwater species. And again, the same problem, like it can infect a large group of fish, like more than 120 different species of fish could be infected by this virus. And it can cause 100% mortality in larvae, juveniles, even in some cases of adults. It was first discovered in Australia, in Asian sea bass. That is Latus calcarifer, which is also present in India. In India, it was reported in 2005. So this virus is categorized under the family Nodaviridae, and it belongs to the genus Beta nodavirus. So it is a non-enveloped virus, and it's spherical shaped, and it is 25 to 30 nanometer in diameter. So there are 5 different genotypes of this virus, and the common mode of infection is horizontal transmission, and vertical transmission is not found until now. And it could, if you see this picture, this is the brain of a fish, and this virus mainly infects the nervous system of the fish. And normally, the infected fish gives a whirling movement on the top of the water surface and eventually die. And this outbreak is also temperature dependent, and it normally occurs during summer season. And recently, ICR-SIBA, that is Central Institute for Brackish Water Aquaculture from Chennai, they have developed a vaccine, recombinant vaccine called NodaVac for this virus. And this more focus have to be given upon this virus, because recently mariculture is expanding in India. Like we are the government and even the farmers are putting a lot of emphasis on mariculture. So in the next decade, for sure, there will be an expansion in the mariculture. So we will be growing large volumes of fish inside the sea. So when the mariculture expands, we can expect outbreak of this particular virus. So more research focus should be given upon this virus. And the next important virus is tilapia lake virus. I'll be spending more time on this virus because it's one of the it's a recently discovered virus, and uh, it was first discovered in Israel, and recently we found this virus in India, and we could find a lot of positive samples of tilapia lake virus recently in southern parts of India. We are getting a lot of samples, so I'll be focusing more on this virus. So if you see the history of this tilapia lake virus, uh, so this virus was, as I said before, it was first discovered in Israel. So you can see the Sea of Galilee. So Sea of Galilee in Israel, it's around 166 kilometers square, and they used to catch mangrove tilapia in the Sea of Galilee. So in 2005, the production was 316 tons. And all of a sudden, in 2009, the production dropped to 8 tons. And there were large losses of farmed tilapia nearby the Sea of Galilee. And even the hybrids of tilapia were dying. And for the next 4 years, they could not identify what was the causative agent of this mass mortality of tilapia. So in 2014, they found this was a novel RNA virus. It was a new RNA virus, and they named it as tilapia lake virus. And immediately, FAO, it is the Food and Agriculture Organization of United Nations, they issued a warning to the member countries, and they gave a special alert for this virus. And similarly, World Health Organization, they published a technical disease card, as well as they started evaluating this virus, how the virus spreads, and they classified this virus as an emerging virus. And similarly, NACA, the network of aquaculture centers in Asia Pacific region, they issued a warning for this virus. So, so the countries which are marked red are the countries which have reported tilapia lake virus. And first, it was in 2014 from Israel, followed by in 2017, a lot of other countries reported that they have tilapia lake virus, like Egypt, then Colombia, Ecuador, then Thailand, Philippines, and in India, we found this virus in 2018. So it is a quite new virus, and the official name is tilapia lake virus. And during initial days, there were like people were publishing in other names like senescial hepatitis of tilapia and 1 month mortality syndrome. And similar to other viral diseases, it was this virus is also temperature dependent. Particularly in Egypt, they could this outbreak happened during summer, during severe summer. So there is a strong. Scientists believe that there is a strong relationship between higher temperatures and outbreak of this disease. So it is a RNA virus, single-stranded RNA virus, and it is 55 to 100 nanometer in dia. And if we analyze the genome of this virus, many parts of this virus are quite unique, and they are not homologous with any other known viruses. And that's why they have proposed a new genus Tilapiani virus and species Tilapiani virus. So it is still under proposal, and this name is not yet finalized. So this is the electron microscopy image of the virus. You could see the viral particles which are marked with the arrows inside the liver tissue. So so still a lot of research is needed to understand this virus. Still, we do not know how long it could survive outside the host, like how long it could survive within the pond in a dry pond or something. So a lot of research is needed on this virus. And what we know is there is a horizontal transmission. So horizontal transmission means it can transmit from one fish to another, like horizontally. If you cohabitate infected fish with a non-infected fish, the non-infected fish gets infected. So there is a possibility of horizontal transmission. As well as vertical transmission. Vertical transmission is from the mother to the offsprings. So if an infected mother is there, and if we produce seeds or if we produce young ones from the infected mother, all the young ones get infected. So there is both horizontal transmission as well as vertical transmission. And So the main organ for infection is liver, but it can also infect kidney, brain, gill, gastrointestinal tract, spleen, and muscle. And we have two whole genome sequence of this virus, one from Israel and another from Thailand. So susceptible species. So it can infect all tilapia. Like there are, there are different species of tilapia and different hybrids of tilapia. So it can infect almost every species of tilapia. However, there was a report from Malaysia that it could also infect tinfoil barb, but still scientists say that that result has to be re-validated. It's not sure whether it could infect tinfoil barb. And it could also infect, the most important thing is it can infect every stage of life, like from fertilized egg, yolk sac fish, fry, fingerling, juveniles, and adults. And people also tried cohabitation experiments. For example, scientists tried whether the infected fish could transmit the virus to other species. For example, in Israel, they were trying with common carp and gray mullet. However, the virus could not get transmitted to common carp or gray mullet. Similarly, in Egypt, the virus mullets were not infected. Similarly, in India, we did a experiment whether the virus could infect our Indian fishes, particularly the Indian major carps, Katla, Rohu, and Nipah. Luckily, the virus could not infect the Indian major carps, milkfish, or pearl spot. Interestingly, pearl spot is also a cichlid. Tilapia is a cichlid, belongs to cichlid, as well as similarly, pearl spot is an indigenous cichlid. The virus could not infect our indigenous cichlid. So that's a good news. So this is a typical clinical sign of the disease, loss of scale, skin erosion, hemorrhages, exophthalmia, abdominal swelling, and scale protrusion. So the social economic impact of this virus. So still now there is no proper research study to estimate the national or the global context of this disease. However, if you see the graph, we can see a large-scale production drop of tilapia. In 2005, it was 316 tons. However, in 2009, it was just 8 tons, and after that, still it's lesser than 2005. So this has affected the livelihood of the fishermen who were, or fish farmers who were dependent on tilapia. Similarly, in Egypt, they had a loss of about 100 million US dollars. So, so a lot of research focus is needed on this particular virus. And the next virus I would like to discuss is Red Sea Bream iridovirus, which is also known as RSIV. It is also a OA listed pathogen, and it is a double-stranded DNA virus. It can infect more than 30 different species, particularly perciforms and pleuronectiforms. And it was first identified in 1990s from Japan. And it's quite prevalent in the Southeast Asian countries. But in India, it was reported last year in 2020. It was quite a new virus to India in the Asian sea bass, that is Latus calcarifer, cultured Latus calcarifers from open sea cages. And the mortality will be from 0 to 100%, because sometimes the host can sustain this virus, viral infection, and they can spread to other susceptible species. And this virus is also temperature dependent and it happens when the water temperature rises about 25 degrees Celsius during summer. It belongs to Iridoviridae, and genus Megalocytovirus. So at the bottom, you could see the viral electron microscopy image of this virus. It is an icosahedral virus with 200 to 240 nanometer, and it's a double-stranded DNA virus, and its genome is it's around 110 kilobase pairs, and it could infect all stages of life, and it can transmit both vertically and horizontally. And scientists think that this virus might have existed in India long back, but it have been overlooked. Like people failed to notice this virus long back. And another important virus is ISKN, which is infectious spleen and kidney necrosis virus. And this virus is very important for aquarium trade. So this virus also belongs to Iridoviridae and genus Aclocytovirus. And the most important thing is like it can infect a wide range of species, like more than 50 different fish species. And another important thing is it is transboundary in nature and can spread by live ornamental fish trade. Like recently, after globalization, we are the world has become more interconnected. We are trading live ornamental fishes between countries. So this particular virus gets transmitted through these live ornamental fishes to different geographical locations of the world. And last year we had 2 outbreaks in Kerala, and it was reported in India in 2020. So right now we are working upon whether this ISKNV infect our indigenous fishes. Like what will be the impact of this virus on our indigenous fishes, our flora and our fauna, our fish fauna. And the next virus is cyprianid herpes virus 2, and it can cause high mortality in goldfishes up to 50 to 100%. And it was reported in Japan long back in 1992, and it has been reported in 16 countries across the globe. And it was reported in India recently in 2016. It belongs to alpha herpes viridae and genus Cyprinivirus. It's also an icosahedral virus with double-stranded DNA, and genome is a little bit large, 290 kb. It's an enveloped virus. So it's highly pathogenic to goldfishes. So goldfish, goldfish, like it's quite common in aquarium trade in India. So a lot of farmers are getting affected because of this virus. It has both horizontal as well as vertical transmission. That means from fish to fish as well as from mother to offspring. And it's also slightly temperature dependent. When the water temperature fluctuates, the fish gets more stressed. And if the virus is in the latent state, that means in a dormant state, it gets activated and affects the fish. And it is a serious threat to Indian ornamental fish trade and fish aquaculture. And next, we'll be discussing about shrimp diseases. Like apart from fish, shrimps are also an important commodity in the Indian aquaculture, particularly in terms of export market. We have a very large export market for Indian shrimps in Western countries. So the most important pathogen was WSSV, white spot syndrome virus. It is a very serious pathogen. So within 3 to 10 days, almost the complete shrimp stock will die if get infected. So a lot of research were done on this virus for the past 20 years. It is a well-studied virus, and it is a double-stranded DNA virus with 300 kilobase per genome. And it is a very unique virus. It belongs to Vispovirus and family Nemaviridae. And you can see the shrimps having white spots on their carapace. So this is a typical clinical sign of this viral infection. And it has a very wide host range, like 120 species. So a lot of other crustaceans like crabs or some aquatic insects can serve as a carrier for this virus. So they can spread this virus from one farm to another, or one pond to another. So so it's such an important virus, and it can infect all stages of shrimp. And it was first reported in China in 1992. Within 4 years, it reached India in 1996. And in 2015-17, we found a new strain of this virus in India. And still, the original source of this virus was a mystery. No one knows where it originally originated. And in early 90s, there was the shrimp aquaculture was a very big. It was quite famous, particularly among corporate companies. Like a lot of corporate companies like ITC, and many other companies invested in shrimp farming in India. Because of this virus, all of them shut down their farming activity and left shrimp farming business. So it's such a deadly pathogen. And next is the monodon baculovirus. And this virus do not cause acute mortality or death in the shrimps, However, it reduces the productivity, like productivity of the overall productivity of the shrimp aquaculture. So this virus belongs to Bacloviridae, and it's also a double-stranded DNA virus with 80 to 160 kilo base pairs, and it was originally from Taiwan, and it said that it's enzootic to wild penates. That means in Indo-Pacific region. If you see the Indo-Pacific region, most of the wild shrimps will be positive for this particular virus, and it can also infect freshwater prawns, and it has both horizontal as well as vertical transmission. And the next virus I would like to introduce is infectious hypodermal and hematopoietic necrosis virus. So it causes a disease called runt deformity syndrome. If you see the picture down, the shrimps have a lot of deformities and irregular growth. And it was discovered, it is a quite old virus. It was discovered in early 90s in the American region, like particularly in Hawaii. And it was reported in India in 1998. It's a small virus, like 20 to 22 nanometer, and non-enveloped icosahedral virus. It's also a DNA virus, and it can cause mortality up to 90% particularly in post-larvae and juveniles. However, adults do not die because of this viral infection, but you can see a lot of deformities in the adult shrimp. And it also have both vertical and horizontal transmission. And the next virus is hepatopancreatic parvovirus. It was first discovered in Singapore in 1982, and it was reported in India in 2002. So it's also an icosahedral virus with 22 to 24 nanometer, and its genome size is around 6 kb, and 4 different whole genomes are available. And the 4 strains are like South Korean strain, Thai strain, Indian strain, and we also have an Australian strain. And it can affect the mid-juvenile stages of the animal. It can cause about 50 to 100% mortality in 3 to 4 weeks. And the main problem with this virus is it causes stunted growth in the shrimp. So even if you feed more, the shrimps don't grow. They'll be stunted. So you'll be having a lot of economic loss due to this stunted growth. It has both vertical and horizontal transmission. And the next virus is infectious myonecrosis virus, also called as IMNV. It's also an icosahedral virus, and it's a non-enveloped virus with the 40 nanometer in diameter, and it's a single non-segmented double-stranded DNA, and its genome size is around 8.2 kilobase pairs. It was first reported from Brazil in 2002, and it was recently reported in India in 2017 after the NASPAD project. It can infect all stages of shrimp, and you can see a cooked appearance on the tail region of the shrimp. So this is the typical clinical sign of this disease, and it can cause mortality up to 40 to 70%. And this virus also have a horizontal as well as vertical transmission, and this is a OE listed pathogen. And next is enterocytosome hepatopenia. So this is a very important pathogen in recent times. So it's not a virus, it's actually a parasite. It's a microsporidian parasite, and in recent times a lot of research have been invested on this particular parasite. And the typical clinical sign of this parasitic infection is we could see a lot of white fecal strings on the pond bottom, and the gut of the shrimp displays a white discoloration. It causes lesions in the hepatopancreas, and it was first characterized in Thailand in 2009. It's a quite recent parasite infection. And in India, we found this parasite in 2016. So the main problem is it causes severe growth retardation in affected shrimp. So the shrimp won't grow beyond a point, and even if you feed more, they won't grow. So if you see the shrimp aquaculture, the most of the money we spend is for feed. So even if you feed more, the shrimps, if the shrimps do not grow, means you'll be losing a lot of money on the feed cost. So a lot of research focus is given upon this parasite, and this parasite have a very thick wall surrounding it, That's why it's very hard to eradicate from the shrimp ponds. So right now, farmers are applying lime, that is calcium carbonate, at the rate of 6 tons per hectare to inactivate this parasite. So this is the structure of the parasite, having a polar filament inside its uh, structure, and it's an electron microscopic image. And so this is the various sizes of shrimp from the same pond stocked at the same time. So there will be a lot of size variation. So if you like in the shrimp export, if you there is a thing called count. So the price of the shrimp is determined by the number of counts. For example, lower the count, better the price. You can explain in such a way that in 1 kg of shrimp, if you have 20 shrimps, it's called as 20 count. If in 1 kg of shrimp, if you have 40 shrimps, it is called as a 40 count. So bigger the size, you get a better price. So lower the count, you get a better price. So this affects the count value of the shrimp, this particular pathogen. And the last pathogen I would like to discuss is white-tail disease of freshwater prawn. And this disease was first discovered in French West Indies in 1999. In India, it was found in 2004, particularly in Macrobranchium rosenbergi. And this is caused by a virus called the Macrobranchium rosenbergi noda virus. Along with this virus, interestingly, there was a satellite virus associated with this virus. It's called an extra small virus. And satellite virus is something called as a satellite virus depends upon the mother virus for some of its replication activities. So we can say like both of these viruses cause white-tail disease in freshwater prawns. And it's a small icosahedral non-enveloped virus, and it's quite small, 26 to 27 nanometer. And in recent times, the aquaculture of freshwater prawn has reduced a little bit, and the outbreak of this disease has reduced compared 10 years back. So next I will be discussing about the National Surveillance Program for Aquatic Animal Disease, which is also called as NASPAT. So seeing the outbreak of these diseases, the government of India started a project called NASPAT in 2013 for screening various emerging disease or screening for new pathogens or transboundary diseases. And this project happened because we made an amendment in our one of our acts, that is prevention and control of infectious and contagious diseases in animals. 2009, we made an amendment in 2014. So we also have, India has an international obligation to screen for pathogen and keep it open for the global community. So so that we can do a transparent trade between our member countries or our countries where we export our fish and fishery products. So we started this project in 2013, and if you see, we have almost covered the whole of India. It's a pan-India project, and the red dots were our laboratories working on this project. It was initially implemented in 14 states of the country, and later it was expanded to 19 states across 2 union territories. Even we have a lab working in Andaman Nicobar Islands, screening for emerging pathogens. And we have 31 collaborating centers, and the institute where I work, that's the National Bureau of Fish Genetic Resources, is the coordinating institute for this project. So what we actually do, we do surveillance. Surveillance in the sense, we have 2 types of surveillance. It can be an active surveillance or a passive surveillance. So in case of passive surveillance, what we do is we look upon literatures or for any for already existing pathogens. Similarly, we will be looking upon outbreaks. Like if a farmer gets a disease outbreak, they can report to us, and we will be screening for the pathogen. And similarly, we we do an active surveillance. We go for a targeted surveillance. We randomly go to farmers, collect samples, and test for already known pathogens. So we have designed this project in such a way that we will be doing both passive as well as active surveillance. Once we get a new virus or a emerging virus in India, then we will be reporting it to our nodal center, and we'll be reporting it to the government of India, and government of India will report to the international agencies like OAE and NACA, and they'll be issuing guidelines to control this pathogens. And second thing is the information flow of disease reporting system in NASPAR project. So if a disease occurs in a farm, it will be immediately informed to the district fishery officer, and at the same as well as the fisheries department will be getting a notification. Similarly, the referral labs will be getting the samples for testing. So we'll be testing the samples. If we find something significant, some new emerging disease or something like that, immediately we will be validating this virus once again in another lab, and we will be reporting to the government of India, and the government of India will again report to OAE. And we will be also issuing advisories to the farmers how to control the pathogen. So all these things are interconnected. And the next thing is, what happens if we find a new OAE or a NACA listed disease, like if it is something new or not found before. So what we do is immediately we will test the pathogen in the lab, and after that we will, as I said before, we will be cross validating in 2 other collaborating labs. So we should not sometimes in lab we get a false positive results. The virus may not be present, but it may give a false positive because of our handling, improper handling in the lab. So we used to send the samples to 2 other labs across the country and cross validate whether the virus is present or not. And again, as I said before, then we inform to the competent authority, and it will be listed in the and the information will be sent to OAE or NACA after that. And we will be issuing advisories, and will be the state fisheries officials will be alerted, and we will be issuing advisories for controlling the outbreak of the disease. So these are some of the new viruses or emerging exotic or emerging viruses not reported in India earlier, but after the implementation of NASPAR project, we like they were found in India, like cyprinid herpes virus that infects goldfish, carpedema virus in koi carp, tilapia lake virus in tilapia, ISKNV in many of the ornamental fishes, red sea bream iridovirus in Latex calcarifer, and we found a bacteria candidatus actinoclamia pangasiae in pangasius, then infectious myelonecrosis virus, and microsporidium parasite, enterocytozoon hepatopeni, and we also found a protozoan, OA listed protozoan parasite from green, which can infect mussels, Perkinsus osploni. And at the same time, we also found that like previously, a lot of diseases were suspected in India. Like there was a confusion among scientists and researchers that some of the exotic pathogens could actually present in India, but no one knew that whether they are actually present or not. So we did an active surveillance to screen for active targeted surveillance to screen for some of the parasites, some of the viruses and pathogens, and we found that those pathogens were not found in India. Like these are the list of viruses, like important exotic viruses, but they were not found in India. Like Vibrio parahemolyticus affin strain, it is a particular strain of serious concern in shrimps. Then koi herpes virus, then spring viremia of calves, IPNV, VHSV, yellowhead virus in shrimps, Tara syndrome virus in shrimps. And if you see, most of these viruses are OA listed pathogens, which are quite serious. And what are the tools used in disease diagnosis? So the commonly used tools are the classical histology. So histology is quite classical technique, and we'll be using histology. And most commonly we'll be using PCR, polymerase chain reaction, and most of the research scholars attending this program will be using PCR. And we also use a multiplex PCR, which is testing two viruses in a single PCR reaction. Then we also do a nested PCR, RT-PCR after COVID. Most of you may be knowing what is RT-PCR. We will be doing it particularly for RNA viruses and real-time PCRs, and we also use DNA-based hybridization techniques. And we are developing a field-based technique called LAMP, loop mediated isothermal amplification, which can be done at farm level. And for bacterial identification, we are using MALDI-TOF. And recently, a lot of research focus is given upon next generation sequencing for sequencing the whole genome of the virus or the whole genome of the pathogen to understand it better. And the researchers are using nanotechnology and nanosensors for detecting disease, like gold nanoparticles for developing instant diagnostic kits. And we are also using various immunological techniques like ELISA, lateral flow test kits. So a lot of research, these are some of the molecular and immunological tools which are used in disease diagnosis, and a lot of research is invested upon these techniques to to make the diagnosis much more affordable to the farmers. And next is the factors influencing the emergence of aquatic diseases. Until now, it was like we were discussing about a lot of virus. It was a little bit boring. Now we'll be discussing about. We can go a little bit more colorful and interactive. What are the factors which, what are the factors which infect these disease? How they are spread to different parts of the world, and how can we control them? So the first important factor is fish trade. If you see, this is global fish trade. So it's highly interconnected. We are exporting a lot of fish and fishery products to different parts of the world, and this has made the spread of disease much easier than before. So 20 years back, we were not trading that much. It was not interconnected much. Now it is much more connected. It has become like a global village. So a lot of viruses take this opportunity to spread across the globe, particularly in case of live ornamental fish trade. The virus like ISKNV spread through live ornamental fish trade. And so we have to, fish trade has to be regulated, particularly in terms of issuing green certification. A lot of screening has to be done before trading the fish. So that can reduce a lot of viral spread across countries. And second thing is climate change. Climate change influences the emergence and spread of this virus. If you see, when I explained about the individual viruses, most of these viruses are pathogens. They are temperature dependent. So when the climate changes, the outcome of the disease can be quite different. Already laboratory studies suggest that we for sure will be having a negative impact when the climate change occurs. So it affects both host and the pathogen, and most of the diseases can favorably spread when the temperature increases. So a lot of research focus should be given upon the interaction between climate change and spread of aquatic diseases. And next is biodiversity loss and diseases. So in recent times, due to a lot of anthropogenic activities, we are losing our biodiversity. So scientists are suggesting that biodiversity losses can also lead to the spread of these viruses. So if a particular animal is removed from a diverse environment, that particular animal could be a factor that could be reducing the infection rate in that particular ecosystem. So if you remove that particular animal, the spread of the disease becomes much more easier, and it could affects the whole system. So a lot of focus should be given upon biodiversity losses, particularly when the biodiversity losses occur. There will be a lot of spillover. Spillover is nothing but when the pathogen spills between wild to culture or from culture to wild. Particularly due to biodiversity losses, a pathogen which is already present in the wild could easily spill over to a culture environment. And similarly, if we don't follow proper biosecurity measures in our farm, like if we don't treat the cultured water uh, and let the water inside natural environment, the virus or the pathogen could easily spread to the wild, and there can be a mass outbreak in the wild environment. So these spillovers are quite common if we don't follow strict biosecurity protocol, or if there is any if there are any biodiversity losses. And the next important thing is antimicrobial resistance. And this is in recent times, this is a very important uh, topic, and a lot of focus have been given by World Health Organization for antimicrobial resistance. So for the past 20-30 years, we are indiscriminately using a lot of antibiotics, not only for humans, but also in the veterinary medicine as well as aquaculture. So whenever a bacterial infection happens, we don't test the bacteria, and immediately randomly we apply a overdose of antibiotics. So this has caused a lot of resistance among the bacterial community uh, for some of the third generation antibiotics. So many of the antibiotics are not working. So this antibiotic resistance not only affects the aquaculture industry but also the human health. So we consume the fish, and we also get the bacterial flora present in our body also get resistant to these antibiotics. So this is a antibiotic resistance spread. So this resistance can get transmitted from, not transmitted, get transferred from animals to humans through food, and even the manure from the cow may be containing antibiotic residues, and those residues will develop drug resistant antibiotic resistant bacteria in the soils of the uh, agriculture fields, and again through food chain they will come to humans, and it's a highly interconnected web. So a lot of in recent times, in the last 5 to 10 years, a lot of research focus has been given upon this antimicrobial resistance. So this factor also causes a lot of emerging diseases. And next is research gaps. So what are the research gaps we face in controlling these emerging diseases? The first one is improved diagnostics. Most of these diagnostic techniques are expensive, so it is not affordable to the farmers. So a lot of research is needed in improving the diagnostics, particularly to make them cheaper for the farmers. And we need to study the genetic variation in the pathogens. In recent times, you might have learned a lot of things about COVID. The virus get mutated, and a lot of new strains develop from the COVID virus. So something similar happens with the aquatic viruses too. So a lot of research have to be invested upon studying the genetic variation of the pathogen, and we should study the host range. So what are the species it could infect, and we should also study the survival, like how long the virus could survive outside the host, and we should also study the environmental factors influencing the pathogen and the host, and we should also develop a cost-effective vaccine for these pathogens. One important thing is we could not develop vaccine for shrimps, because shrimps have a totally different immune system than fishes, because they don't have an adaptive immune system. That means they don't produce antibodies. So the concept of vaccination do not work for shrimps. However, in fish we can develop vaccines for fish diseases. And we can also test our archived samples. For example, the labs will be storing their old samples in formalin or in minus 80 degrees Celsius. So we can go back to our old samples and test whether these viruses present in our environment long back, and whether we might have overlooked these viruses. And research collaboration. And to control these emerging diseases, we need a research collaboration between different states as well as international community, and we need to develop resistant strains of fish and specific pathogen-free strains of fish, and so that needs a lot of research in, and it's developing a resistant strain would be a huge investment. So we need an international collaboration for that, and there should be a public-private partnership. The farmers should there should be a good relationship between the farmers as well as the research labs, and we should also invest upon import risk analysis. When we import a live fish, we should screen for the viruses. Proper quarantine should be done, and we should continue the surveillance process. So if we do a proper surveillance, we get an early alarm for any new viruses, so that we can prevent new diseases. And we should invest a lot on understanding the new strains of the virus. And prevention and control strategies. How can we prevent? So the same thing again. So we should develop a resistant strain, and we should develop strict biosecurity measures, particularly by certification, like issuing disease-free states when we import. And even if we import, we should have quarantine facilities. We should keep the fishes in quarantine and observe for the clinical signs of disease. And we should have a farm level biosecurity. Every farm should have a strict biosecurity measure, so that even if an outbreak happens, it do not spread to the wild or other farmers. And vaccines, as we should develop vaccines, and we should develop SPF broodstock, that is specific pathogen-free broodstock. We should have good management measures. We should practice good management pressures at the farm level, and we can use information technology. We can combine information technology for controlling disease. Particularly, we can use early warning systems, like a well-connected system between the researchers as well as the farmers, and we can map the disease incidences using GIS mapping. And we can a lot of in Europe, people are going for predicting the virus now. So they have records for 20 years of disease outbreaks at various seasons of the year, and they have developed predictive modeling. So the model will predict, you can expect a particular disease outbreak at this season. So we should invest a lot of research on predictive modeling as well. And that's all from my part, and thank you so much for listening.
Thank you so much, sir. Sir,
hello.
Some of the questions in the chat box, sir.
Yeah. Okay.
So the first one, sir, which condition?
Which condition should be considered in viral
nervous necrosis challenge, sir?
Condition in the sense, I don't understand. Like condition in the sense, like you mean the temperature, or?
Yes, sir. Some of the factors.
Yeah. In case of viral nervous necrosis, temperature plays an important role. So you mean if you want to conduct an it depends upon the research objective, actually. So for example, if you are interested in the influence of the temperature on the viral infection, then you should consider about temperature. But normally the experiments are done at around 27 degrees Celsius. Most of the experiments are done at around 27 to 32 degrees Celsius. But temperature, of course, temperature influences the outcome of the result. So higher the temperature, the outcome of the disease will be different.
Okay.
But it totally depends upon the research objective, what the researcher want.
Okay.
The second thing,
Second one is why epizootic ulcerative syndrome is called as a syndrome or not the disease.
not a disease. So this is an interesting question because for a quite long time people were not knowing the causative agent of the disease. It was I think it was almost 10 years, like it was a quite long time, like more than 10 or 20 years later they found out it was Aphanomyces invadens, and even after that there were a lot of debate that whether it was actually caused by Aphanomyces invadens or it was caused by some other virus. So that's why it was called as a syndrome. But still, people are using the same old terminology, episodic ulcerative syndrome.
What will be the efficient way of therapy with the chosen antibiotic that is in the treatment process?
Again, this is a very important question, particularly with regard to antimicrobial resistance. So, with with my personal experience, like uh, recently, most of the antibiotics are not working for bacterial infection. So the best thing to give a therapy is to do an antibiogram test, like testing the, so we can isolate the bacteria from the infected fish, do an antibiotic sensitivity test. So in that you will know which are all the antibiotics which are resistant to the pathogen, and you can use the antibiotic which is sensitive. Sensi- which actually acts on the bacteria, and you can use that as a therapy. And we can either directly mix in the water, or we can mix them in the feed and give them. The problem with the feed is most of the infected fish do not consume feed, so that will be a challenge, but we can mix in the water.
Okay, so. And one more thing, which environmental parameters in water quality could be considered as causative agents in occurrence of enteric red mud disease, that is in rainbow trout?
Actually, when I was doing my PhD, one of my other colleague was working on enteric red mouth disease. I do not have much knowledge on this particular bacteria. However, temperature was an important parameter. Like temperature was really an important parameter for the outcome of the disease.
Okay. The last one, can parasites be presented as bioindicator of water quality and aquatic ecosystem health?
Yes, for sure it could be. Considered as a bioindicator of water quality. Even not only the water quality, even the outcome of the disease. For example, I was in my PhD. I was working on a myxozoan parasite called Tetracapsulotus bryosalmonae. It was a myxozoan parasite. So in the wild, in the natural environment, there was a strong relationship between eutrophication of the water and the parasitic infection. So whenever the eutrophication was a little bit whenever it was on a higher side, the infection rates were more. So for sure we can correlate between parasitic infections as well as water quality. And sometimes parasitic infections totally depend upon temperature. Most of the protozoan parasites infect when there is a fluctuation in the temperature.
Okay. Thank you so much, sir, for your enlightened talk on the diseases of fishes and how to prevent the diseases. Yes, sir. Thank you so much.
Thank you so much.
Yes.
I thank the organizers for inviting me to this national seminar, and it was really I really enjoyed it. Thank you.
Yes, sir. Thank you, sir.
Hello, sir.
And
I have one small doubt, sir.
Yeah.
I am from C. Abdul Hakim College. We are the part of National Repository Program.
Yes.
For the first time, we have isolated many viruses that IMNV, new strain of WSSV, WTD.
Yeah. Most of the work which I presented were from your group actually.
Yeah, thank you. I have one small doubt that is there is any control measure for EHP?
Um, right now, like I know from the farmer's perspective, like people are mixing vinegar, little bit of vinegar in the feed and feed them and feed shrimps, and farmers are saying they are getting better results from that. And similarly, a high quantity of lime is used after between culture period to inactivate the spores, EHP spores.
I can discuss with this chat for Kusti.
Yeah.
Yeah, that we are working on nodoviral infection, that is viral nervous necrosis.
Yeah.
Here, temperature plays a major role.
Yeah.
Where we can propagate the nodovirus till 4 degrees Celsius. Recently, we established the rainbow throat cell line. There we are conducting that temperature plays a major role in nodoviral infection. And regarding that episodic ulcerative syndrome, it is due to the multiple infection of episodic Aphanomyces invadens, Aromonas hydrophila, and snakehead rhabdovirus. Here we are targeting to isolate the snakehead rhabdovirus using the snakehead cell line. And yeah, that's it from my side. We are maintaining all the positive control. If any student needs any positive control sample for their research work, they can request us with an official letter so that we can provide the sample to them. And also, we are the national repository for this cell line. We can provide the cell line also. Okay, thank you.
I have a question: whether viral nervous necrosis can infect? You're working on rainbow trout cell line?
Yes, yeah.
Okay. It could infect rainbow trout.
Yeah, we recently established 3 cell lines from rainbow trout.
Okay.
And we are working on the rainbow trout cell line.
Okay.
Okay, thank you.
Okay, thank you.
Sir, excuse sir.
Yeah.
2 more questions, sir.
Yeah.
How can aquaculture farmers identify this big arena of diseases?
It's what to say, like it's a big challenge actually. They have to depend upon some laboratories for identification, like most of these NASPAD, the laboratories under NASPAD, they do they test the samples for farmers. So at field level, it will be a little bit difficult. Like it's really you can do only limited things. So they have to depend on some well-established laboratories for that. And we are often like many of these labs are giving trainings for farmers to identify these pathogens at field level, and a lot of research is going on developing a diagnostic kits at farm level. But still, it is a challenge.
Yes.
Yeah, under that surveillance program, we used to go to the farm monthly once, madam. Our detail is available with the farmer. If there is any problem, immediately farmer will inform us so that we will collect the sample, or farmer will send the sample to us so that we can check the infection. That we are doing free of cost under this NASPAD program, National Surveillance Program. That we are doing. Who are the farmer cooperating with us? We are helping them. And recently we developed that field level kits so that farmer in the field they can detect the disease.
Okay. Thank you, sir. Thank you so much, sir. I call upon Nisha Gracelyn of 3rd Zoology to propose the vote of thanks.
No duty is more urgent than that of returning thanks. Good morning to all. I deem it a great honor and privilege to propose the oath of thanks on this pleasant occasion. Above all, I thank the Lord Almighty for blessing us with this day. My sincere thanks to the speaker of the day, Dr. Arun Sudhakar, scientist, Peninsula and Marine Fish Genetic Resource Center, ICA, National Bureau of Fish Genetic Resource, Kochi, Kerala, for his session on present stages of emerging disease in Indian aquaculture. Sir, your talk on the disease of different aquatic breeds and the different dose used in diagnosis and the factors influencing the emergences of aquatic animal diseases were useful. We also gained knowledge about the mariculture and different types of viruses, transmission of co-culture species, and social impact. Thank you, sir, for this useful session and your words of wisdom. I would like to convey my sincere thanks to our Vice Principal, Reverend Dr. Sister Mary Fabiola, for her constant support. I extend my gratitude to IQAC Coordinator and Head of the Department, Dr. Paul and Basanti Joseph, and our dignified faculty members for their immense support and motivation. Before winding up, I would like to thank all my fellow mates for making this even successful. Thank you.
Thank you, Nisha. Thank you very much, Dr. Arun Sudhakar. That was a very in-depth lecture and a very good, vivid description of the diseases in aquaculture that you see, and also the surveillance measures that are taken up by different agencies. Thank you so much, sir. Thank you so much for giving your consent and for being with us here today.
Oh, thank you.
Thank you so much, sir. So participants, we have the last session for today, and this session will be chaired by Dr. Ramamurthy from the CAS of Marine Sciences, which comes from Marangipete. So we look forward to Dr. Ramamurthy's address now. Can we have Dr. Ramamurthy? So, good morning, sir.
Good morning, madam.
Yes, sir. It's so nice having you for the first session of the seminar. But
Nice to
indeed,
share.
it's a very important session. And this is a field that almost each one of us would like to do something about, especially ornamental fishes, and something that is very practical in our labs. So we are most happy to have you, sir. And we look forward to a very resourceful lecture now. I'll just hand over this session to Dr. Divya, Assistant Professor from the Department of Zoology.
Okay, thank you, madam. Screen?
Good morning, everyone. On behalf of the Department of Zoology, Jamala College for Women, I extend a warm welcome to all for the third session on the second day of 4 days national seminar on aquaculture production, advances and constraints. I find immense pleasure in introducing a honorable speaker of this session, Dr. K. Ramamurthy, Associate Professor, CAS in Marine Biology, Faculty of Marine Sciences, Parangipete. Thank you so much, sir, for accepting our invitation and coming over for the seminar to give a lecture on ornamental fish culture sector and overview. Welcome, sir.
Hello.
Hello. Can you hear me, sir?
Hello, madam.
Can you hear me, sir?
Yes, I could hear you.
Oh, okay.
Shall I start my lecture?
1 minute, sir, for the formal welcome address, sir.
Okay.
I now call upon Atulya from 2nd MSc Zoology for the formal welcome address. Atulya?
Yes, ma'am. Am I audible?
Yes, Atulya.
Coming together is a beginning, keeping together is a progress, and working together is a success. With this motivational quote, I wish a great day to one and all gathered here. It is my pleasant duty to bid you all a genial welcome to this auspicious occasion. I feel extremely honored to welcome our resource person, Dr. K. Ramamurthy, Associate Professor, Department Faculty of Marine Science, Annamalai University, Palangipetai. Dr. K. Ramabhuti sir has specialized in marine ornamental fish breeding and have an experience of 18 years in ornamental fish breeding. Sir has established a marine ornamental fish breeding hatchery facility in the Faculty of Marine Sciences, and 12 marine clown fishes have been successfully bred in the estuarine water. And the value addition of fishes' skin color enhancement studies was taken on commercially important fresh and saltwater ornamental fishes. Sir has conducted 5 training programs for coastal villages on the marine ornamental fish aquaculture, and Sir has successfully guided 5 PhD scholars and also published 32 research papers. It's indeed an honor to welcome you, sir. I also extend my warm welcome to the eminent faculty members and all the participants who are present here for this virtual gathering. Once again, I welcome you all for the session. Thank you.
Thank you. Thank you very much.
Thank you, Atulya. Sir, it's over to you for the presentation, sir.
Yes, sir. Thank you. Could you see my PPT?
Yes, sir. It's visible, sir.
Yes. First of all, I would good morning to all, and Principal Madam and HOD Madam Masanta Joseph Madam. Today I'm going to present. So today we are going to discuss about the ornamental fish sector and overall view. So what is ornamental fish? From the basics to breeding, up to breeding. So I will try to, what I am planning to give, some basic knowledge, for the ornamental fish and how to maintain the ornamental fish and where we are getting all the informations. Okay, we will go into the subject. The first of all, the basic question is, what is ornamental fish? What is ornamental fish? Is there any definition for ornamental fish? Yeah, it is. So, if you see the total number of identified species in India, all over the world, it is 34,000 plus in number. It is keep on increasing. It is new species. New species are adding in this list. Definitely, the species, the identified list will get increased. So, if you see these fishes, among these 34,000 plus fishes, how many species are ornamental fish? See, this ornamental fish, number of ornamental fish so far identified is only 1,539 among these 34,000. 1,500 something fishes. Other fishes are food fish. If you classify the whole fishery biodiversity, it is there are 2 types of fishes. One is food fish, another one is ornamental fish. Food fish, you know, what is food fish? What is the definition of food fish? Whatever we eat, that is called food fish. But whatever the ornamental, what is the definition of ornamental fish? It is nothing but unusual color and shape. Unusual color and the shape. You can add the unusual behavior also in addition. So, if you see the ordinary fish shape and size of the ordinary fish, this is the ordinary fish shape and size. See, as a zoologist, it is very easy to understand. This is the caudal fin, dorsal fin, ventral fin. This is the usual shape of the fish. But if you see these goldfish, same caudal fin modified into long size. Long size. This is the anal fin. See, the normally anal fin should be like this only, but in the ornamental fish, anal fin is such a long, and dorsal fin is long. Likewise, then you see this marine. This is marine angelfish. See the shape of the fish. This dorsal fin extended up to caudal fin. So it's a shape. You see the shape also. So what is the ornamental fish? Means unusual color and shape and behavior. This is the criteria of ornamental fish. The food fish color, you see the food fish color, there won't be any color. But color is also important. So unusual color, shape, and behavior is very important for ornamental fish. Next. What is the value of ornamental fish? So I'm comparing with the food fish. What is the value of ornamental fish? If you see the food value of food fish always, like how we are buying the fish, 1 kilo, 1 kilo, 2 kilo, this way you used to buy. If you see the value of ornamental fish, it varies from 100 to 1000 rupees per kilogram. So the sardine fish, sardine fish, it may be 100 rupees per kg. But if you go for sea fish, the maximum valuable, commercially valuable is 1000 rupees per kg. But if you come into the ornamental fish, there is no limitation for the value. Suppose the example, you take this chili arvana. This is the vastu fish, chili arvana. A good quality chili arvana fish in the market, it will be 5 lakhs per piece, not kilo, not with a kilogram. The same way, if you see the marine clone fish, each fish is cost about 500 rupees. Likewise, this fish using it's called flower horn. This flower horn, minimum 5,000 to 10,000 rupees per piece, small one. So if you see this thing value, then the ornamental fishes are considered in higher value when compared to the food fish. Okay. So where we get this fish? What is the fishery resources in the world? If you see these fishery resources, the red color, this is the equator, Bhumidya Rekha equator. From the equator, 25° north and 25° south. This is the tropical region. This is the tropical region. In this tropical region, it is very close to the sun. It is very close to the sun. So we get a very good sunlight. In this area, this area is getting very good sunlight and the temperature and the light effect. Due to this temperature and light effect, the productivity of fishery is very high in this area. So this area is considered producers, and the other areas are not cold country area. They are consumers or buyers. This graph shows the detail of the productivity. So in red color, this scale shows temperature, scale of temperature. The maximum is 30°C, this is red color. You see, this area is getting very good sunlight source. So this area is the highest productive area in the world. So India is locating in the tropical region. India is located in the tropical region. Little bit India, the top of the India is coming under the less temperature, the Himalayan area. Okay. So actually, the temperature is very, very important for sexual maturity. Sexual maturity in the cold climate, it delays the sexual maturity. But the warm water, it accelerates the sexual maturity. So the whole productivity is the major fishery productivity occurring only in the tropical region. Then where do aquarium fish comes? Okay, we are going into the aquarium market or wholesale or retail. Several aquarium fishes are coming. So where they are coming? There are two types of fishes, We are going to study detail about it. Freshwater aquarium fish and the marine aquarium fish. So if you come to the freshwater aquarium, 90% of the aquarium fishes are cultured fish, hatchery produced. Only 10% of the fishes are wild collected from the wild. And if you come to the marine fish, it is just opposite. Only 5% of fishes are cultured, 95% of the fishes are collected from the wild. What is the reason? Here only 10% are from the wild. Here 95% of 90% of the fishes comes from the wild, because in the marine fish we don't have the most of the marine fish. 95% of the marine fish we don't have fish breeding protocol, fish breeding techniques. There are a lot of problem in the breeding problem, because when compared to freshwater fish, the maturity level of the maturity age of marine ornamental fishes it's too late. Sometimes some fishes it takes 15 years, 20 years also. And another one big problem is sex reversal. After maturing, after maturity, the male fish converts, female fish converts into male. So it is a very big issue in the marine ornamental fishes. But freshwater fishes, it is not like that. It is very easy to breed when compared to the marine fishes. That is the issue. So still researches are going on, and in the marine ornamental fish, in near future, we will succeed reproduction of several good species. So most of the marine freshwater fishes are sustainable. Sustainable means we will not disturb the natural environment. But the most of the marine ornamental fishes are still not sustainable. We are depending on wild collection. So wild collection means, see how they are collecting with the scuba diver. Most of the ornamental fishes are coral reef associated fauna fishes. So they use different type of net and the poison and several disturbing method. They are collecting the fishes from the coral. So where do ornamental fishes are coming? Some are collected from the wild. Some are collected from the wild. Some are collected from the farm. These are the hatchery produced, the clone fishes, young ones. So where some are collected from the wild, some are collected from the farm. Okay. Yeah, this is what is the benefit of ornamental fish culture? What are the advantage of ornamental fish culture when compared to the food fish culture? So if you see the freshwater ornamental fish culture, you have to have, food fish culture, you have to have large volume of area, very big pond, very big quantity of water. But the investment also very high. And if you see the culture period, the food fish culture period, minimum it needs 6 months. Maximum it extends 10 to 12 months. And another big problem is variation in market price. Variation in market price. If you see this, particularly in this month also, most of the people will take a non-vegetarian. So immediately the price will get reduced. This problem is there in the food fish. Next one is lengthy maturity period. You have to wait for long period. If you come to the ornamental fish cultures, what is the advantage of ornamental fish culture? Small culture area. We have to have a small culture area is enough. So you need small investment. Definitely small investment. If you see the culture period, maximum culture period is 6 months here. Maximum culture period is 6 months. Here minimum is 6 months, but the culture period is very, very short time in the ornamental fish culture system. Then fixed market price. There won't be that much fluctuation in the market. Then shorter maturity period. So if you see the biodiversity of ornamental fish in India, see already I told that in the world the identified number of ornamental fish is 1,539 species. But if you come to the Indian biodiversity, how many ornamental fishes are there in India? These are the indigenous species. The Indian flag is there. if you see the Indian indigenous freshwater fish, indigenous fish, I know you are as a zoologist, you know very well. Indigenous fish, our native species. Our native freshwater species, 374 species are existing. If you come to the marine, 700 species are identified in our coastal area and our coastal area. But if you see the other, however, 300 exotic marine species and 27 exotic live bearers and 260 species of freshwater exotic fish layers. So these are all exotic fishes. This much exotic fishes we are importing from the other countries. Okay. This I would like to show some commercially important ornamental fishes. This is the commercially important indigenous species. It is collected from India. We are exporting to other countries. So these are the very good species, very expensive and very common species. So this is a zebra species. This is the glassfish. I'm going to tell a story about this fish. And another this is gourami. This is pencil fish. Yeah, this is black shark, beautiful black shark, and knife fish. And this is barb variety. This is also rosy barb, beautiful fish, rosy barb. This is another one, barb fish. This is drawfish. These are all common, very common Indian indigenous species. But the commercially important exotic egg layers, Oscar fish. This is another famous fish. This is African fish. It comes from African country. This is an important shark. This beta fish. This is beta fish, and this is hybrided fishes. And goldfish and chichilids. These chichilids also from South Africa. And exotic, commercially important exotic egg layer fish. Egg laying fish, if you see the egg laying fish, the koi carp is very, very important. Koi carp. And the kissing gourami. And black-tailed shark, neon tetra. These are all common, very common fishes, very important fishes in the aquarium market. Aquarium market. This is also the egg layer, the continuation of the egg layer, discus, arwana, the vastu fish, angel fish, and spotted gourami. Yeah, if you come to the live bearers fish, the guppi, mali, and swordtail platy. These are cheap quality fishes, but very, very commercially important fishes all over the world. Okay. If you come to the global trade of the aquarium fish, if you see the global trade, some 7 to 8 countries are occupying 80% of the trade. United States, UK, USA, Japan, Germany, Netherlands, and France. These countries are occupying this much portion of the trade. But if you see the India won't be there. What is the percentage of India? I will tell you later. This is the top 10 exporting country. If you see the top 10 country, Sri Lankan export portion is 4%. What about India? There is no India here also. Well, how much India's export and India ornamental fishes is a 0.5%, not even 1%. So India name is not coming in this map. So if you see this Asian market, Singapore is number 1. 25% of the ornamental fish trade is trade is occupied by Singapore. But Singapore is not producing that much ornamental fish. Singapore is acting as a hub of world ornamental fish trade. Singapore is buying and selling. Singapore Sunday cholvanga, Sunday. Those who want to buy the fish, you can export it to Singapore. So if you want to buy any fish, you can get from the Singapore. So Singapore is acting as a very big hub for the South Asian countries. Okay. This is the top 5 important country under the percentage contribution. This is the top 10 I told now. This is the top 5. UK, USA, Japan, and German, France. These countries are occupying the top 5 ornamental fish trading. Okay. Now you come to the Indian ornamental fish resources. If you see the Indian ornamental fish resources, see this green color to green color, we have very good river system, beautiful river. We have very big river system, a pond and several very big lake. Here, very big lake. Here, there are several beautiful ornamental fishes are available, but we are not catching and exporting. If you come to the marine ornamental fish, we have very big island system, Lakshadweep Islands and the Andaman Nicobar Islands. If you see the Andaman Lakshadweep Islands, there are 300 species are identified in Lakshadweep, marine ornamental fish. Beautiful marine ornamental fishes. In Andaman, you see 150 ornamental fishes are identified. If you see the Gulf of Mannar near Rameswaram Island, 100 species are identified in this area. Same way, this Gulf of Mannar near Gujarat, the 40 species were identified. But what is the problem? The Indian government completely banned the ornamental fish collection and exporting collection and exporting. That is one of the issue. But if they open the ornamental fish trade, again our greedy fellow people will destroy this coral reef definitely. That is why the government is completely banned the marine ornamental fish export and marine ornamental fish catching and exporting and trading. So if you see this thing, what is the benefit of ornamental fish keeping? So human beings are most selfish animal in the world. So what is the benefit? Without benefit, we will not do anything. So aquarium keeping is said to be the second largest hobby in the world. So what is the first hobby? The first hobby is photography. Next is ornamental fish keeping. So such a ornamental fish keeping is such a very big hobby, not only in all over the world. In the most developed countries are the first is photography all over the world. The second is aquarium keeping. Aquarium keeping is that much important in all over the world. Now we are going to see the few health benefits of aquarium. So if you see the health benefits, the first one is reduce eye exercise. What is eye exercise? If you go to yoga center, they used to teach Angini Kriya. In Hindi word, that is Angini Kriya. Angini Kriya means, what is the left, turn your eyes left, turn your eyes right, turn your eyes up, turn your eyes back, turn eyes rotate, clockwise rotation and anti-clockwise rotation. Now, this is the yoga is daily, doing yoga itself will be stress. But if you keep an aquarium at your home, you don't have to go for these eye exercise. Just watch the fishes for 20 minutes. You will achieve all the exercise. High focus and low focal and the rotation, left side rotation, right side rotation, top rotation, everything. So your whole eye exercise will be done in 10 to 20 minutes. Next is, it is very good blood pressure reducer. Once you watch this aquarium, the fish movement and the fish activities, definitely it is a scientific study. All are scientific study, scientifically proved. It reduces the heart rate and BP, your blood pressure. Those who are having the BP, blood pressure, just you keep aquarium, definitely it gives very good results. Then improve the sleep. The fish keeping is it gives positive energy to your home or bedroom. Definitely, if you get the positive energy, you will get good night's sleep. Okay. Then reduce the pain and the pain and the trauma. That is there is a protocol that all the dental hospitals should be there aquarium. Definitely, those who are feeling this, the dental pain is very big pain. So those who are coming with pain, if they watch ornamental fish closely, definitely they are forgetting the pain. The pain is reducing. That much attractiveness and this thing is the benefit is there in the aquarium keeping. So next important is therapy for hyperactive children. If you see these hyperactive children, they will not sit and do anything. They do some naughty things. So if you keep aquarium at your home, they spend lot of time with their aquarium, feeding the fishes and watching the fishes. What fishes? This thing, the fishes will play with the children. So this is very good treatment for the aquarium. Is very good treatment for hyperactive children. And definitely, if your blood pressure reduces, if your the night sleep increases, the productivity will get improved. And for the children, this is a very good learning opportunity. Very good learning opportunity means the children can understand the ecosystem. What is ecosystem? How fishes work? What is the ecosystem? The ecosystem means what is the interaction between non-living and the living things. So these things very easy to learn from the beginning. So next is aesthetic profession. Definitely, if you have very good aquarium at your home or office or any place, it gives additional look and additional attraction. Okay. Next, we will see a little bit about the aquarium preparation. If you go to the aquarium preparation, if I ask me, there is a definite shape and size. There is no definite shape and size, defined shape and size. That is beyond your imagination. It is beyond your imagination. I'm going to show some very good photos. See, there is no limitations. This way also you can prepare the aquarium. Yeah, this way also you can prepare the aquarium. You can prepare aquarium in your tea table. You can prepare aquarium in a photograph like this. In most of the Kerala hotels, you can see like this aquarium. Yeah, this type of aquarium. This is also a tea table. This is also a tea table. This is the bedroom aquarium. This is half bell aquarium. Half bell. If you in the half bell aquarium, this type of aquarium you can keep it in these beta fishes. These fishes doesn't need any aeration. This is air breathing fish. Air breathing fish. Okay. So this is some crazy aquarium. You see wash basin like aquarium and cylinder aquarium, the commode aquarium, and pot type of aquarium. So aquarium, there is no limitation. This is preparing a small bulb, diffuse bulb. Small bulb, small bottle. So okay. This is another this thing we should not miss this. This is the planted aquarium. So the aquarium, if you see these the ornamental plant are also very important. It is equally important for ornamental fishes. Aquatic plant or ornamental plant. These are called the aquatic plant or ornamental plant. These plants are the submerged plant. These plants are see these settings. See these settings. It grows taller. It grows like Korean grass. It can grow like leaves. So several types, some 150 species are there in these aquatic ornamental fishes. aquatic ornamental plants. So this is the pond, fish pond. In Singapore, Malaysia, most of the Asian countries, these fish ponds are famous. In their backyard, they used to construct fish ponds. Evening time, they spend a lot of time with the fishes. See, this is a one-sided glass and another is covered. Yeah, this is a type of pond fish system. This is the koi fish. This is fish and the plant. This is the mobile aquarium. Okay. So now we are going to see some theoretical part. So aquarium setup. What is the aquarium setup? Based on the salinity, it is classified into 3 types. One is fresh water, brackish water, and salt water. You know what is fresh water? What is fresh water? What is freshwater fish? See, 0 to 10 ppt. Up to 10 ppt, 0 to 10 ppt come under fresh water. Those fishes are living 0 to 10 ppt, that is called freshwater fishes. It is very important. And those fishes are, this is the coral reef fishes, they are very sensitive fishes. They need 30 to 35 degree ppt. If you go for brackish water aquarium, this is a narrow range, wide range of tolerant species. It can tolerate 0.5 to 30, up to 30 ppt salinity. See, this is based on the classification is based on the salinity. Okay. Next, the freshwater aquarium further classified into cold water and tropical water fish. So cold water means this is the world standards. Less than 25 degrees Celsius comes under cold water fish. In Himalayan region, I told you, in our Himalayan region, we have very good cold water, freshwater ornamental fishes. And others are tropical ornamental fishes. Tropical means above 25 degrees Celsius, the tropical water fishes. Okay. The freshwater aquarium, there are 3 types of setups. One is fish aquarium, only fish aquarium. Next one is, I told you, aquatic ornamental plant aquarium. Next one is fish and planted aquarium. The further classification, fish aquarium, single fish aquarium, and community fish aquarium. Single fish aquarium means, in this, you take arowana or these flower horn fish, this you cannot, this you have to grow with one fish in the single tank. If you put another one fish, immediately they will fight and die. So this is see, in the fish community, there are 2 types of friends: friendly fish and enemy fish. In same film, this lives only one in one tank. This cannot adjust with other fishes. These fishes, but these fish, friendly fish, this can adjust with other species fishes. So how to find out these friendly fish and enemy fish? Don't worry about that. This chart is available in the internet. The compatibility chart for the marine freshwater fish. So if you type this compatibility chart for freshwater aquarium fish, you will get it from the internet. So this graph, what it shows, the Y is compactable. You see, suppose the Y, you take Y. This fish is compatible with this fish. This and these fishes are friendly. If you the N, whatever N is not compatible. The C is caution required. You can put the fish, but you have to take care of the fish. If you are not feeding anything, if you are not feeding 1 or 2 days, immediately the cannibalism will be there. Cannibalism, they will fight and eat. So same way, this is the marine compactability chart, marine compactability fish chart for aquarium. Okay. So Suppose if you go for preparing your aquarium or you are going for aquarium business or anything, so you have to know the budget and the maintenance. What is budget and maintenance? So if you see this chart, this is the risk meter. So this is the fish aquarium. This is the plankton aquarium. This is the coral reef, marine coral reef aquarium. So if you get the budget and the maintenance wise, for the beginner, you have to go with this next level, you can go for this. So this is the budget wise, it is very easy to maintain. And the next level, this is budget wise, medium budget and moderate difficulty and moderate risk will be there. This is high risk and high budgeted aquarium. Okay. Suppose you have prepared the aquarium. So what is this? What are the species? How to select the species? For the beginning, the aquarium, there are 3 types of fishes are there. Easy to grow. These fishes are very easy to grow. Goldfish, koi fish, guppi. These fishes are very easy to grow. Suppose if you are preparing a new aquarium, first you have to start with this type of fishes. This type of fishes easy to grow. What is the easy to grow means? What is the meaning of easy to grow? They are hardy fishes. They can tolerate any narrow range of water chemistry. This can tolerate any hardy species. This can tolerate any shock. This is a medium cat species. This is a medium cat species. It is not that much easy, but not that much difficult. This is also medium cat species. This is difficult to grow species. Difficult to grow. What is the difficult to grow species? Because of their aggressiveness. You know, the piranha, this jellyfish, this is a highly carnivore fish. If you don't feed one or two, even one day, immediately one fish will eat another fish. This is also difficult to grow, these fishes, but because of their delicativeness, it needs with, I told you, the easy to grow species, this is just opposite to this species. This species cannot tolerate any water chemistry changes. Any water chemistry, even in a small oxygen level reducing or pH change, any change, it cannot tolerate any water chemistry changes. It is very difficult to grow this species. So, what is the average life of different ornamental fish species? If you see the life of the fish, you should know now all these things. Suppose what is the average life of human being? What is the average life of your dog? What is the average life of the ornamental fish? If you see the ornamental life of the ornamental fish, if you see this betta fish, the life is maximum 2 to 3 years. But if you come to the goldfish, the life is 10 to 30 years. A goldfish, what is the life of goldfish? 10 to 30 years. Okay. Now we will go into the fish breeding. Fish breeding. Everybody knows the fish breeding. This is the general life of a fish. What is the general life of the fish? There will be a male and female parents. They lay the egg. Female lays the egg, and male injects the milk, and it will get fertilized. The fertilized egg will hatch out the larvae. The larvae will develop as a fry. After fry, it will reach in the fingerling stage. What is the fingerling stage? What is the fingerling is? Up to this larval and the fry stage, you cannot identify the species. It will not, in this stage, the fish will not look like adult. Once it reaches the fingerling, this fingerling will look like adult. So this stage is important stage. That stage is called fingerling stage. Then juvenile, then adult. So this is the cycle. This is the cycle of a fish. This is the cycle of a fish. But we are going to see detail in the marine ornamental fish breeding. If you see the marine ornamental fish breeding, based on the culture method, there are three types, basic types. One is freshwater egg layer. Egg layer. Egg layer, the name itself, it lays the egg. Next one is freshwater live bearers. Live bearers means it produces direct young ones. There won't be any egg laying. And another one is marine species. This is the major three divisions. Okay. But here we are going to see detail about the freshwater freshwater fish reproduction. If you see the based on their reproductive pattern, it is divided into two types. One is live bearer fishes, another one is egg laying fish. If you see the live bearer fish, it is classified into two types. Live bearers. What is the live bearers? What is the live bearers? Live bearers, they produce direct young ones. Egg layers, it lays the egg and it hatches. Okay. Live bearers, it is again classified into two types, ovoviviparous and viviparous. Ovoviviparous and viviparous. The egg layers, the egg layers is further classified into egg scatters. Egg scatters means, what is the meaning of scattering? Just it will scatter the egg. Female will scatter the egg into the open water. The same time, the male will scatter the sperms. So the fertilization will happen in the externally, and the parent fish will not take care of their eggs. Only the releasing itself is enough. They will not take care of their eggs further. Next one is egg depositor. They will not scatter the egg. They will deposit the egg on rock or some sticky substance. They may take care of the egg, and they may not take care of the egg. There are two types of that. Next one is nest builders. They build the nest like birds. They take care of their egg till hatching. Next one is mouth brooder. Mouth brooder. The mouth brooder, they take care of their egg and larvae in their mouth even for 15 to 1 month. They will not take any food. The mother fish will not take any feed. After reaching the fingerling stage, then only they will release the young ones out. In this mouth brooder, there are two types of, again it is classified into two types, oophilic mouth brooder and larvophilic mouth brooder. So I'm going to show all the photographs, don't worry about it. If you're not understanding this thing, the next slides you will understand clearly, don't worry about that. The mouth brooders, again it is classified into oophilic mouth brooder and larvophil mouth brooder. Oophil means just the fishes lays the egg outside. Immediately all the egg will be collected by the mother fish, and they will keep into the mouth. The larvophilic mouth brooder is, they lay the egg outside. The egg will be there in the outside. After hatching the egg, the young ones will be kept in their mouth. So the mouth breeder is again classified into two types. Okay. So what is the general steps for fish breeding? For fish breeding, if you see the fish breeding, both live bearers and the egg layers, there are 5 steps. The step 1 is the setting up the spawning tanks, spawning tank and the spawners, mother fish setting. Next one is selection of spawners, selection of good quality spawners, original species and good quality mature spawners. And the next one is conditioning the brooder fish. Conditioning the brooder fish means we have to give special food for mother fish, brooder fish. So mother fish. The next one is spawning. Next one is spawning, means spawning is nothing but releasing the egg and sperm. That is very important stage. Next one is rearing the fish larvae. You have to save the fish larvae. Fish larvae. Okay. So just I would like to little bit. I would like to give little bit detail for this oviparous and the ovoviviparous fish. What is the, actually I told you, the ovoviviparous and viviparous fish, they directly release the young ones. It means we should not think about the human birth. What is the difference between, it is also, see, the fertilization the fertilization is internal and development takes place in the mother body of the mother stomach. But it is not like a human fertilization. Human fertilization is completely internal fertilization. The baby is connected with the mother with the umbilical cord. But here there won't be any connection with the mother and the baby, mother and the baby development. Then how babies are developing? The babies are developing with the egg yolk only. There won't be any connection with the mother and these young ones. After the development, the young ones released to the genital apparatus of female. This is the definition of live bearers. Live bearers, they directly produce the young ones, but it is not true internal fertilization. There is no umbilical cord. These fish, when the matured female fishes produces the eggs. The male will injects the sperm into the egg. The egg will fertilize and develop inside the mother's stomach. They will release. But it is not true internal reproduction. Okay. What is the live bearers? So this is the first we are going to see the live bearers reproduction. Next we are going to see the egg layer reproduction. The live bearers reproduction, if you see the live bearer reproduction, I'm going to show the cartoon for the live bearers reproduction. But this is the theory part. You see, one is first you have to separate the male and the female. When you are after purchasing the brooder fish, you have to separate the male and the female in a separate tank. Feeding with this variety of healthy food, even for the healthy food. Healthy food means pupa content. You have to give the pupa high polyunsaturated fatty content feed. Then only the number of egg and the quality of sperm will get increased. Then feeding with the live. Then what are the feed? Feeding with the live feed. Brain shrimp, Artemia, insect larvae, and the earthworm. These things are. These things will give very good polyunsaturated fatty acid for mother fishes. Make them in suitable condition for spawning. We have to prepare suitable conditioning for spawning and reintroduce the fish. Reintroduce the male and female fish. The fish will be ready to spawn. Male and female you separate for 15 to 20 days. Reintroduce. After reintroducing, immediately it will spawn. The spawning will occur. After spawning, larvae, you have to collect the larvae. After collection, the larvae, you have to give larvae live feed for larvae also. After larvae collection, you can go for maturation, earthen pond maturation. So this is also detail about the live bearers. Already we have seen. Already I told all those things. This is there is no here you see. There is the umbilical. There is no umbilical cord. Okay, that is very important. So this is the general for your information. I am giving how much the fecundity of ornamental fish. Fecundity means number of egg releasing capacity. If you see this is a very small fish. It will look like very small fish, but the gabby maximum it can lay 100 eggs. The platy is also small, very small fish, but it can produce 500 number of eggs. The molly, this 100 eggs, and the swordtail, this can release 200 number of eggs. 200 number of eggs. So the 200 of this for your example, I cannot give. See, if you see the ornamental fish breeding, this is not general. There is no. We cannot categorize general single methodology. See, if you see the method of protocol for ornamental fish breeding, each and every species has separate breeding protocol. So, just I have generalized for your reference. I am giving some single. This is the example of example species I am giving. If you see the goldfish fecundity is 5,000 eggs, 5,000 number of eggs. If you see the koi fish, it can lay 1 lakh, 2 lakh, 5 lakh eggs, 5 lakhs eggs also. So depends on each and every fish, the number, the fecundity number, and the breeding behavior, and breeding behavior, and the fish standardization. It differs fish to fish. So this is the general life cycle of a guppy fish. You see, this is the day 1, day 30, day 60, day 90. Within these 3 months, from larvae to the maturity period, you see the maturity period of a guppy fish 90 days only. After 90 days, again it can go for breeding. So this is so I told you the life cycle of each and every ornamental fish, it varies. Next is egg layer. I told you egg scatters, egg depositor, next nest builders, and mouth brooder. If you see these things, the parental carries keep on increasing. Egg scatters, in these egg scatters, there won't be any parental care. Just the parents will not take care of their eggs. But egg depositors, it will care little bit. Nest building, it is the care will be there for 50%. And the mouth brooder, 100% care will be there. You know what is the parental care? As a zoology student, you know very well. What is the highest parental care animal, Highest parental care animal is monkey. The monkey, it takes 8 years parental care. After 8 years, only this monkey baby will go alone and take their food. Up to 8 years, they used to keep with the mother. Okay. The egg layers, the egg scatters. The egg scatters, what is the egg scatters? The egg scatters, they simply scatters their egg in the open water. There are 2 types of eggs in the egg scatters. One is adhesive egg and not adhesive egg. Adhesive egg means there will be a coating on the egg. There will be a coating on the egg. They will go and stick on somewhere else. But non-adhesive egg, there won't be any coating. There won't be any coating. It will float on the open water. There are 3 floating methods is there. floating method. One is surface floating egg, and another one is mid-water floating egg, and another one is submerged eggs. They do not look after the brood. They will not, those fishes, the scatterfish, they do not look after their brooding. They even eat their own eggs. Their fish group their pairs. After the large number of small eggs, okay, this much is enough for what you have to understand. What is the egg scatters? The egg scatters is they scatters the egg in the open water. There are two types of egg scattering. One is adhesive egg and non-adhesive egg. This is the non-adhesive egg. This photograph you see, this is floating egg. This is adhesive egg. It will stick on somewhere in the rough substratums. This is the egg depositor. The egg depositor example, you take clone fishes also egg depositors. They deposit the egg. The female lays and stick the egg in the rough substratum. The male will follow the female and will spread the sperm on the egg. The egg will get fertilized. Till fertilizing, the parents will, both parents, male and the female, will take care of their eggs. This is the mouth brooder. All the cichlids varieties and arowana fishes are mouth brooder. I told you now two types of mouth brooder. The ovophilic mouth brooder. This is ovophilic mouth brooder. Ovophilic mouth brooder. The egg, they lay the egg. After immediately they will take after laying the egg, they will collect all the egg in their mouth till hatching and till the young fingerling stage. They keep in their mouth. But arowana, you see, the arowana is coming at a larvophilic mouth brooder. They hatch the egg. After hatching, they collect all the egg in their mouth. This is the egg yolk. The next very good example is beta fish. This beta fish, this is the egg. If you see from the aerial view, top view, it will be like this only, some foam-like substance. They produce some foam-like substance in the water. You see, this is the underwater. This is from the aerial view. This is from the inner view. If you see the inner view, they will stick on their egg in the foam. Egg in the foam. This is a gourami. This is a beta fish. They will deposit the egg in the under this foam. Till hatching, they used to take care of their egg. After hatching, they will all the young ones will go out from this foam. Okay. So this is our marine ornamental fish breeding. My best friend, Dr. Ajit Kumar, he only initiated this ornamental fish breeding. He successfully bred 14 species of ornamental clone fishes in our laboratory. I'm continuing the same breeding in our department. See, this is the brooder. And these are young ones. This is our native species, Amphibirian seabae, clownfish, Amphibirian seabae. This is another one species, Oscillaris. These are the famous ornamental fishes. This is ornamental. This is clownfish. This is கடல் தாமரைன்னு சொல்லுவாங்க. கடல் தாமரை. Sea anemone. Carpet sea anemone. கடல் தாமரைன்னு சொல்லுவாங்க. Carpet sea anemone. This is the tentacle sea anemone. This is another one species, Amphibirian platypula. See, this much young ones. Okay. Actually, Dr. Ajit Kumar, now he's continuing the same research in Mumbai also. Okay. So this is very important for student point of view. This is the value addition of ornamental fish. What is the value addition of ornamental fish? You know, actually, if you see the goldfish, this is the original goldfish. From this original goldfish, this much varieties of colorful goldfish have been developed. What is the technique? You know, several techniques are there. We are going to see. See, this is the original koi fish. From this koi fish, this many variety colorful fishes were developed. This is barbel. This is original barbel. These are all developed varieties. This is the glass fish. This many varieties. This is zebra fish. This is the original variety, but we have developed this many varieties. What is the how to? This is the fighter fish. In the fighter fish, if you see, this is how many colorful fighter fishes are there. How to develop these colorful beta fishes. So see, there is a program gene jumping. What is the gene jumping? Gene jumping is nothing but there is a chromosome, movable chromosome. This is a movable chromosome in the beta fish. Just if you move the chromosome to another place, immediately you will get some different fishes. That is called the gene jumping process. By gene jumping, you can develop different kinds of variety of colorful fishes in beta. Beta fish, it is very successfully working. Next one is artificial coloring. I told you now glass fish. Glass fish is nothing but colorless transparent fish. In this transparent fish, by using these colorful dyes, colorful fluorescent dye, by colorful dyes, you can convert into beautiful fishes. Beautiful fish this way. See, by using the small injection and the dye, you can inject the color dye into the fish. Immediately the fish will get change like this. See how many varieties of shark, colors shark, like tetra fish, bar fish. By adding different colorations, what we are using this thing tattoo, tattoo dye. Tattoo dye is mixed with ethanol or something. It is secret. Still, it is secret. We cannot do easily. It is not easily. It's not that much easy. Some special enzyme and solvent they are using. So how to create the new species? How to create the new species? You know, hybridization and the crossbreeding. This is very important technique for producing the new species. Next one is the fluorescent transgenic species. The third one is, I told you, the important this thing is induced breeding. Induced breeding is also very important for increasing the production rate. The third one is, I should not forget, this ornamental aquatic plant. I told you, the ornamental plant, the ornamental fish plants there are several ornamental plants are available in the world. It is expensive. It is equally expensive with the ornamental fishes. You see, there are a lot of ornamental fishes are there. This is also a very big field. Actually, before going to the tank, so for your convenience, for easy understanding, I have prepared a small cartoon for the freshwater ornamental fish breeding and live bearers and egg layers and marine ornamental fish. See, this is a step one. Then the step one, you have to collect the brooder, good quality brooder, male and the female fishes. Next, you have to separate the female and the male for 15 days. For this is the step two is conditioning. I told you now, conditioning for the breeding. The third one is reintroducing and spawning. So you take one male and one female. So 6 tanks you have to have. Here you have to begin with one large big tank, then 2 small tanks. One is for female and one is for male. Then you have to have 6 tanks. Each female, one male. You have to keep. So if you keep these things, the spawning will occur. Female will lay the egg, male will release the milk. So after releasing the egg and the milk, you have to remove you have to separate out the male and the female parent fishes. After collection, after removing these things, you have to give live feed for young ones. That live, what is the name of the live feed? Infusoria. The culture of infusoria, it is very easy. While adding this fish, you have to put some vegetation, waste vegetable, potato or tomato, some brinjal pieces. From this waste vegetable, automatically the infusoria will develop. Infusoria is nothing but the microorganisms, the microscopic live feed. So the microscopic automatically it will develop in the tank. So all the young ones will feed in the micro infusoria for 1 or 2 days. Then you have to go for feeding the fishes, pellet feeding the fish. Then you collect all the larvae in a big tank, that is larvae culture. Then you have to go for pond culture, rearing the outside. Then you have to go for packing and selling. Next is the freshwater ornamental fish culture, live bearers. How to culture the live bearers? Same way you have to collect the parents, male, female parents, and separate the males and separate the females. This is conditioning the fish for 10 to 15 days. Then you have to go for breeding. Then you have to go for breeding. Male and female. The male, after these things, it will release the young ones directly. Then you remove the young ones and you can go for pond culture and stocking and packing. So this is the marine ornamental fish. Marine ornamental fish is a very typical characteristics. I will tell you. This is the broodstock. Broodstock, if marine ornamental fish are crowded in a group, all the fishes will be intersex. This is the intersex. There won't be male or either male or female. It will be intersex. Sex will not be determined. But what you have to do, you separate out one small fish and one big fish in a tank. Immediately, what will happen? Immediately the gender will occur. One will become female and another will become male. One will become female and another one will become female. Then you have to give some hiding substratum for fish and laying the egg also. So the first process is you have to collect this brooder fish. The brooder fish, if the brooder fish are intersex, there won't be any female and male separation. Then gender will occur. Next, after conversion of female and male, always the male will be small, female will be big. So after after this conditioning, they lay the egg in the pot. After 7 days, it will hatch out the larvae. After the metamorphosis, you will get small young ones. This is the culture method. So here you see culture period. You see the culture period. This conditioning will take 20 to 40 days minimum. The conversion male to female, female to male. So sorry, intersex sex determination will take 20 to 40 days. Then the hatching of egg, after laying, hatching of egg will take 7 to 80 days. The larvae culture will take 60 to 90 days. 60 to 90 days. This is the culture period. The culture setup, first you have to have a brood stock, then pair formation, and the egg laying hatching capacity section, and the larvae culture, and the grow out. In feeding system, if you see for the mother feed, you can give fish meal and meat. But this stage, the feeding, live feed for parents, rotifer for young ones. Here you have to give live feed, only live feed, the young ones. Live feed, artemia or pellet feed. So this is the detail of marine ornamental fish breeding technique, particularly clone fish. So these clone fishes are not sex determined. They are born male, but they can turn into female at some point of their lives. So this sexual reproduction is called clone fish or sequential hermaphrodite sexual system. Okay. Suppose if female, suppose this male fish dies means you can add another one fish. If you stock another one fish, that will this new fish will convert male. That is the advantage of clone fish aquaculture system. Okay. Thank you very much for listening. Thank you very much for listening.
Thank you, sir. Thank you so much, sir, for making an excellent presentation. And sir,
if you have any doubts, please you can ask.
Yes, sir. Now the session is open for questions. Girls, you can post your questions or you can unmute and ask yourself. Sir, there is 2 questions in the chat box. So what is the percentage of protein, lipid, carbohydrate and other additives in ornamental fish culture?
So, madam?
What is the percentage of protein, lipid, carbohydrate and other additives in ornamental fish culture, sir?
Actually, if you see the fish, each and every fish for the growing, suppose your feed preparation, if you see the poultry culture, or if you egg layers, if you go for egg layer variety, you have to give more protein. If you want, likewise for larvae culture, you have to give more fat content food. But if you go for the mother fish, brooder fish, you have to give more protein content feed.
So next question, sir. Can I read it out, sir?
Hello?
Can I read the next question, sir?
Yeah.
So what is the water quality requirements for fish culture, sir, for ornamental fishes?
For ornamental fish culture, I told you, for ornamental fish culture, if you go for freshwater fish culture, those fishes from 0 to 10 ppt, up to 10 ppt living fishes are coming under freshwater fish. So I told you, each and every fishes has their own breeding protocol. Suppose you take an example, goldfish. Goldfish, it needs 5 ppt salinity. 5 ppt salinity. So each and every fish has their own protocol for water quality. But general water quality, freshwater water quality, what is the water quality for fish breeding? It should be, it should not exceed anything, any extremes.
Sir, one more question, sir. So the color change of the ornamental fishes, is it due to the genetic modifications or is there any other reason for it, sir?
Color change, whatever color change is, any color change?
Color change of different ornamental fishes, is it due to genetic modifications or is there any other reason for it?
Yeah, one is genetic modification, and another one is transgenics and color injection, and another one is this thing. You can promote the color by giving the fish color is controlled by the fish the skin melanin. That melanin is controlled by, as there is a chemical astaxanthin. By giving the different astaxanthin, you can change the color or improve the color. These astaxanthins are available, different astaxanthins are available in the chemical laboratory, or it is available in the market. Astaxanthin.
Thank you, sir. Is there any other
Hello,
question
sir.
from the
Hello.
Sir, good afternoon.
Good afternoon.
So I have a question. Like you were telling about how these danureras and tetras were being injected with some dyes to make them colorful.
Yeah.
So in the are they able to breed on their own, or like everything is artificially inseminated?
But artificially, what is the artificially inseminated? But
Like
that is the temporary process. The color will not be permanent. It will stay for 1 or 2 months only. After that, see the fish body automatically it will metabolize the color. Any chemical substance, if you inject any chemical substance into the fish, the metabolism, after metabolism, immediate permanent process. This is the purely temporary process.
Okay, sir. And also, I've noticed that in my local aquariums in my city, these aquarium or ornamental fishes, the death rate is higher. Is it because they are not maintaining it properly, or because of all these different colors or dyeing, they tend to have a very short lifespan?
Actually, madam, see, if you see, what we are living is air medium. We are living in the air medium, but the fishes are living in the water medium. So the water medium means if there are any issues in the air medium, it is very easy to escape. But fishes are living in the water medium. If any changes in the water quality, immediately the fish will get response. So the water quality is very, very important. Yeah, the water quality means it is very not easy to tell about the water quality, because if you see the water quality list of this thing, there is a 240 water quality characters are there. So it is very difficult to assess the water quality also. What happened in the water?
True, sir. Thank you so much.
Sir, I have one doubt, sir. If our students or scholars are willing to become an entrepreneur in ornamental fish culture, what is your suggestion or idea to become a successful entrepreneur, sir?
Yeah, okay, madam. So first you have to have a certificate. The certificate says that you know something about the ornamental fish culture. Next, madam, in the central government, MPEDA, Marine Export Authority, this thing, and the NFDP, National NFDP. This NFDP and MPEDA, they are giving lot of loans for the fish culture, fish keeping, ornamental fish shop, everything. They are giving minimum, they are giving 50% subsidized loan. Suppose if you get 15 lakh rupees loan, only you have to pay 7 lakhs only you have to pay. 7 lakhs will be subsidized. For that, there is a procedure that you have to fill up. Suppose you are going to start a farm, you have to have own land or lease land for lease for 5 years lease agreement. So these type of small conditions they have. But very easy to get the loan and very easy to start this business. The MPEDA and the NFDP is supporting like anything for this ornamental fish sector development. There is a scheme in MPEDA, Rainbow Revolution. Rainbow Revolution. Under the Rainbow Revolution also, there are several type of loan they are giving. They are giving 50% subsidy for this ornamental fish sector. Any this thing, you can go for fish hatchery or fish rearing or aquarium shop, anything. They are giving 50% subsidies loan. Another one, PM scheme is there. PM, so exactly I don't know, but my phone number is this. Please call. I will tell you all the detail. The Prime Minister scheme also, they are giving special fund for this ornamental fish culture, backyard ornamental fish culture. That is also 50% loan. That for SC they are giving special discount.
Yes, sir. Thank you, sir.
Okay.
Thank you so much, sir, for your detailed explanation.
Thank you.
Is there any more questions from the participants? Okay, I now call upon Kalai Archana from 2nd BSc Zoology for a formal vote of thanks.
No duty is more urgent than that of returning thanks. So first, I thank God for blessed all us with good health. A grateful and warm afternoon to our most valued guests, worthy teachers, and most beloved my friends. Last but not least, all gathered over here. It is my privilege to have been asked to propose on this occasion. I extend a very hearty thanks to our chief guest, Dr. K. Ramamurthy, Associate Professor, CAS in Marine Science, Parangipettai. Thank you, sir. Your session was an eye-opener for ornamental fish aquaculture sector about benefits of ornamental fish and aquarium sector in aquarium budget, maintenance, and many important things about fish breeding steps. And your session was very interesting and useful for us. Thank you, sir, for sharing your valuable time with us. If you want to turn your life around, try to thankfulness. I extend my gratitude to our chief patron, Reverend Dr. Sister Ruby Alangara Mary, who always motivates us. Thank you, sister. And I heartily thank our principal, Reverend Dr. Sister Helen. She motivates us in all possible ways. Thank you, sister. I thank our Vice Principal, Reverend Dr. Sister Mary Fabiola, a kind-hearted, and she encourages in all good times. Thank you, sister. The special thanks for our Dr. Fazin Vasanthi Joseph ma'am, HOD of Zoology Department, for organizing a great national seminar for us. Thank you, ma'am. And my wholehearted thanks for our dignified staff members for the immense support you rendered to make this occasion successful. Finally, I thank all the participants for making this seminar a successful. I thank all the people who have been the backbone of this event. Once again, I thank all for your cooperation. Thank you.
Thank you, Kalai Selvi. So, Professor Ramamurthy, thank you so much, sir, for giving your consent. And that was a very wonderful lecture, very pleasing to the eyes and pleasing to the mind also. And I think you had a lot of questions from the audience also. Thank you so much, sir, for being with us.
Thank
you
you.
very much, sir. Thank you, sir.
Thank you. Thank you.
The participants, the feedback link is provided in the chat box. You can fill the feedback link. So we keep the platform open for 10 minutes. Please fill up the feedback link for your certificates.